Okay. So
log in kora na. Log in kora.
So very good morning.
Vikram, please.
Vikram, you switch off your microphone.
So a very good morning and a warm welcome to the Symposium Molecular Dissection of Human Diseases. Today we are celebrating our director, Professor Soumitra Das's contributions to science, as well as his 60th birthday. I would request now our founding director, National Science Chair and Distinguished Professor, Professor Partha Pratim Majumdar, to kindly deliver the opening remarks.
Partha, please unmute yourself. Partha, please unmute yourself.
Yes. I'm so sorry. Yeah, I muted myself. Yes. Good morning, everyone. All of us have assembled here for a special symposium on molecular dissection of human disease. And this is in celebration of Professor Soumitra Das's 60th birthday. We're just about a week late. His birthday was on 20th January. He was born on 20th January 1962. It also coincides with a somewhat sad note in the sense that Professor Soumitra Das is leaving us with an uncertain future. He has been steering the institute for the past 3.5 years, and we've gotten to know each other, and we knew what his plans for the institute were. But now, for reasons that are beyond his control, beyond our control, he has to leave and go back to the Indian Institute of Science, where he is a senior professor, and we have no choice but sadly to have him leave us. But again, that leaves us with an uncertainty. We don't know who is going to take the reins of this institute. But be that as it may, we are very happy to celebrate his 60th birthday, and with a whole one-day symposium that has a number of people who have been associated with him in various ways. I just want to recall a few things about Professor Soumitra Das, and essentially I'll confine myself to the academic areas in which he has contributed. He got his education in Calcutta. He did his PhD from the Indian Institute of Chemical Biology, and then did a postdoc at the University of California at Los Angeles. He returned to Calcutta in 1998 and assumed the role of the position of an assistant professor at the Indian Institute of Science in the microbiology and cell biology department. For his work, he quickly rose to the ranks of a professor, and was also instrumental in initiating and coordinating a center of excellence for research on hepatitis C virus. This was, of course, funded by the Department of Biotechnology, and that center was one of the stellar centers of the Department of Biotechnology. Professor Das has contributed widely, mostly in the area of molecular virology or molecular biology of viruses. He initiated this kind of work right from his doctoral thesis, where he looked at interactions between the pathogen Leishmania donovani and the human host. It causes Kala Azar, as many of you know. So early on in his career, he started looking at various kinds of pathogens that cause human disease. So it's only appropriate that we are today celebrating his 60th birthday with a symposium that's titled Molecular Dissection of Human Disease. During his postdoctoral at UCLA, he identified a small RNA molecule from the yeast, Saccharomyces cerevisiae, and demonstrated its antiviral activity against polio and hepatitis C viruses. The next many years saw him contribute in an immense way to human hepatitis C virus. And in his independent research, this is where he has made most of the contributions, scientific contributions. He identified and worked out the molecular biology of factors that bind to HCV, internal ribosome entry site, and influences internal initiation of the hepatitis C virus. He showed the importance of competition between host and viral factors that contributes to the molecular switch from translation to replication of the HCV RNA. He worked out the molecular biology of these interactions and discovered that some microRNAs and long non-coding RNAs play important roles in these interactions. Currently, he's involved in the characterization of some of the relevant miRNAs in HCV life cycle and pathogenesis. It causes, as all of us know, hepatitis C virus induces hepatitis in the human. It causes human disease. And the insights that Professor Das gained from molecular biology of the host pathogen interaction enabled him to identify those proteins that serve as antivirals. He also worked out possible modes of delivery of these antiviral proteins. He then turned his attention to investigating whether some herbal compounds could target HCV proteins and work as antivirals. Indeed, he was able to find some and showed that these were efficacious, non-toxic, and bioavailable. His work on antivirals naturally led him to vaccinology and led him to ask whether he could find a HCV vaccine to prevent HCV infection. His work in this area has provided some early and successful leads that are being tried out in animal models, primarily the mouse and the pig. I think that work has kind of taken a back seat since he moved to Calcutta to take on the reins of our institute, but I'm sure that it will gain enormous momentum once he goes back. Professor Das also has worked with other kinds of viruses, notably the Coxsackie virus, B3. I won't dwell very much more. His work on on the hepatitis C virus has been phenomenal, has been groundbreaking, and overall his work on host-virus interaction has been profound and potentially of huge translational value. Professor Das has mentored a large number of students and postdocs. He's developed collaborations within and outside of his own institute. Professor Akhil Banerjee was a collaborator who was outside of his institute, and the two of them made some contributions, deep contributions in the area of host pathogen interactions. Professor Das has been awarded with many honors and medals. He had received the National Bioscience Award for Career Development. He was elected a fellow at different points of time in his career of all of the 3 science academies of India. He received the Nasi Reliance Platinum Jubilee Award, the Ranbaxy Award in Medical Sciences. He was also selected as a J.C. Bose National Fellow in 2014, I think. So overall, his career has spanned a long and deep contributions to science. And he has made a profound he has contributed to profoundly to the understanding of human host and various kinds of pathogen interactions that have led to finding of treatments or preventives, and also exploration of various kinds of vaccines. We were very happy and lucky to have had him as our director for the last 3.5 years, and he has steered the institute. When he came to the institute looked very different, and as he's leaving now, it's gained momentum and it's gained new heights because of his tireless, dedication to steering the institute. As I said, we are sorry that he has to leave us now, in the sense that he goes back to his parent institute and continues to contribute to science, but we will miss him because we won't have his leadership and stewardship that he provided in the last years. Aside from scientific contributions, many of us also know that Professor Das is a Rabindra Sangeet exponent, and we hope that sometime during the day he will sing a few Rabindranath Tagore songs for us. We certainly expect that from him as a parting gift. So without further ado, let me welcome all of the speakers of today's symposium, the molecular dissection of human disease. Again, I remind you that this one-day symposium is being hosted by the National Institute of Biomedical Genomics in honor of Professor Soumitra Das. I hand over the microphone to the compere of today's program. Thank you very much for your attention.
Thanks very much, Professor Majumdar. Now I would request the chairperson, Professor Sharmila Shendra, to take over and initiate the symposium.
Thank you, Anushwa. And I also extend a warm welcome to all of you who have joined in the audience, and welcome the speakers as well. And regarding Professor Das, I do not have much more to say because Professor Majumdar has already covered extensively his academic career, his contributions to science. But indeed, I join my voice with him that we are sad that he is leaving us. I wish he would be here for some more years. Then the institute would have been much more in better shape. But it is already, it looks very nice. As you step into the campus, it's a beautiful campus now that we have. And all this is because of Professor Das's initiative, as well as the labs are also enriched with various kinds of facilities with genomics added on to our genomics facility as well, and zebrafish and model systems. And we are also trying to set up an animal house along with ISAR Kolkata. It's almost done, and I'm sure it will be functional, but I sincerely wish that Professor Das would come back and still provide his drive and initiative and encouragement to the scientists working in NIBMG. And today we will not lament because he's going away. This is a part of his life, and we are here to celebrate his 60th birthday as well as to honor him because of his contributions, immense contributions that he has made in science to science and society. So today's symposium is the brainchild of essentially the faculty members of the National Institute of Biomedical Genomics. Some of us, we got together and we decided that we will, although it's named molecular dissection of diseases, we thought that we would cover the areas and we would try to highlight the areas in which Professor Das essentially worked. Therefore, we have invited Professor Akhil Banerjee, who is a virologist and is also has been a collaborator of Professor Das. We invited Professor Sushant Roy Choudhury. Given that Professor Das is also interested in, he works on certain aspects of P53 in cancer pathogenesis. And we have with us Professor Umesh Varshney, with whom he is not only his colleague from Indian Institute of Science, but as I'm sure they have worked together in the area of RNA biology, contributed immensely together. And we also have with us from our institute, we have Dr. Sridhar Chinnaswamy. And Sridhar is also a virologist and an immunologist as well. So we essentially try to cover what Professor Das was interested in and still pursues. So with this, we will now move on to the symposium because it's a tightly packed program. And I would like to introduce now the first speaker of today, Professor Akhil C. Banerjee. Professor Banerjee did his PhD did his work at the National Institute of Virology and got his PhD from Savitribai Phule Pune University. For his postdoctoral work, he went to the US and did his research at Duke University Hospital, as well as at the National Institutes of Health. On his return to India, he joined the National Institute of Immunology and chaired the Department of Virology at a point of time. In between, he had a 2-year stint at Colorado State University as a visiting professor, I think during the period 2002 to 2004. And Professor Banerjee also served as the director of the Institute of Advanced Virology in Kerala for a period of time, but now he continues to be at the National Institute of Immunology as an emeritus professor. His research interests are in the use of lentiviral vectors, stem cells, catalytic nucleic acids, which are ribozymes and DNA enzymes, and small interfering RNA technology as a gene therapy approach against HIV and other pathogens on one hand, and to determine the functional implications of genetic changes in the host genes or viral genes from HIV-1 isolates in order to understand the molecular basis of HIV/AIDS pathogenesis in India. He has published high-quality papers in reputed journals, and he has been awarded by the Department of Biotechnology the National Bioscience Award for Career Development. Besides, he is also a fellow of the National Academy of Sciences, India, and the Indian National Science Academy. Besides being the recipient of the National Foreign Associateship of the Department of Biotechnology and the Shakuntala Amit Chand Award of the ICMR, that is the Indian Council of Medical Research. We are honored to have Professor Banerjee with us, and he will be presenting his work on exploitation of host cellular factors in modulating HIV-1 pathogenesis. With this, I request Professor Banerjee to share his slides and start making his presentation. Professor Banerjee, please.
Yes. Can you see my slides, please?
Not yet.
Not yet. Okay, then what do I do? 1 second.
Sir, switch on your mic, camera, and then share your slides.
Okay, I have to go back again then. Let's see. I have to switch, turn camera on.
Yeah, we can see you, sir.
But you are muted.
You have to unmute yourself.
Is it?
Yeah.
We can hear you. We can't hear you.
Professor Banerjee, you muted yourself again. You need to unmute yourself.
I think maybe.
Yeah. Now, again it's going.
Maybe better to log off and log in again.
Is it unmuted now?
Yes.
Okay, now I go to slides?
Yes, please do.
Oh, I haven't pressed.
Also.
You have to share your slides.
Okay. Let me go back. I have to. Go to share, right? Open share tray, huh? Then share slides, huh?
Yes.
Share screen.
Screen share.
Share screen.
Okay. Can you see now?
No.
You can
Not
see?
yet come.
To switch on your camera.
Let's see, share. Oh God. Oh God. Share entire screen. Oh God. Yeah, I switch camera
Yeah,
on.
yeah. It's coming.
Camera
Yeah.
on.
Yeah, it's coming now.
You can see the slides?
Yeah, I can see the slides, sir, but your mic, just switch on your mic. Camera, sorry, camera. Camera.
This is the one.
Uh-huh.
Can I go to slides now?
No, just now he has come, but his camera is not working, I guess.
Yeah.
Sir, your camera is not working, I guess. So you can start now, sir. We can see your screen.
You can see, but
I
okay.
cannot.
You can see the screen?
Yeah, minimize it. Please minimize it. Yeah, now.
You can see the screen?
Yeah, we can see.
Okay. Can I start now?
That time now it has come.
Can I start?
Yeah, yes, sir. Please start.
Press presentation mode.
Just make it full screen.
Make it full screen, Professor Banerjee.
Yeah.
Yes,
Good.
it's perfect.
Okay, perfect. So, good morning. And first of all, my very sincere thanks to Dr. Partha Majumdar, we call Parthoda, and for giving me this opportunity to be a part of this celebration, which is 60th birthday of Soumitra Das. And so, Soumitra, a very happy birthday to you and many more happy years and a very fruitful and very significant research that you've already been carrying. So I'm sure you will continue with a similar enthusiasm at Institute of Science, Bangalore. But before I go on further, I wish to accord my very sincere appreciation about the vision and the brainchild that of this institute, which is Partha Majumdar, Dr. Partha Majumdar. And so we are all, the nation is grateful to you for all the services this institute is providing. This is really world class, and in terms of genomics and health. And it has already been helping so many people, and it has made a mark in the global scenario. Okay. So now to talk about Dr. Soumitra Das, yes, I have known very closely Dr. Soumitra Das as my colleague, I can call him a younger brother. And I have watched his science very closely, and I was wondering how his basic science, the remarkable feature of his basic science had been to his ability to exploit the application part of it. Okay, this is where it distinguishes his work from many of the virologists. In fact, I would rank him as one of the top virologists of this country. So, am I being heard clearly? Hello?
Yes,
Yes, sir.
Yes.
Okay. So to say he has made very Parthoda has covered everything about his contribution. Okay, but very few things that I would like to add is yes, he has contributed very significantly in the hepatitis C biology and many other viruses, along with his latest interest in P3 and cancer. So those have been very justifiably covered by Parthoda. And yes, played a very key role in promoting science everywhere, and especially, as Dr. Partha said, about elevating the levels of this institute. So we are truly very thankful to him for all his dedication and service. Okay, so and besides that, I've shared some meetings at Molecular Virology. Oh, he's credited with starting Molecular Virology Forum in India, and many task force members and other academies. Okay, and he has been particularly helpful in my ability to select some faculty for my institute, which I've just left actually. So besides that, before I go on to talk about my work, there are some very, as Parthadas, I've not known that he's, although it is implicit that any Bengali would be a worshipper of Rabindra Sangeet, and we would definitely, this is something which I had not known this part of this side of him. And so the other third, that he's a foodie, just like all Bengalis are foodie, besides relishing food. I just want to give one small incident. Once we were together in Bombay, and then it was a lunchtime and we were out in a taxi. So the taxi fellow, or we did not know where to go, but Soumitra Das knew precisely where you could get very good pav bhaji in Mumbai and which place. So he took us to that place. So this is just one example, how Ganatantrik Soumitra Das has been with food. Okay, so I stop here talking about Dr. Soumitra Das, and now let me begin my talk about my work at NIV, National Institute of Immunology. So I joined my institute in '94, and then I was working on HIV at NIH, and I started working. So this is one very colorful slide you may have seen quite some time. All that I want to show the student, that if this is, let us say, whatever this color is shown here, blue or green, blue, here it is blue, and India is also completely. So we have in India, we have subtype C. Okay. So, but it all started in Africa. Africa is most colorful. It started then in the subtype B, US and UK, and many recombinants. And let me tell you one thing. We were first to show that. Let me talk about this one. CRF, complex recombinant form, isolated in 2002, and it is AG recombinant. Okay, this was usually found in Central Africa, and we reported for the first time in India. Anyway, so this is just a complex recombinant. Complex recombinant can again recombine. So as has been the case with many RNA viruses, they can recombine and they can make mosaic viruses. Okay, all the time. Now, so for students, if they are there in the audience, we have subtype C, even 90%, but we see increasing BC recombinants. There's a remarkable protein called HIV-1 TAT. Okay, TAT is the one that drives the engine of HIV. In a HIV-1 replicating cell, supply amount of STAT, and then millions and millions particles will come out. So that is the wonder molecule of TAT. TAT also, I and Ranga, we showed that we see BC recombinant in the TAT gene. Okay. So this is the structure, gag ball and envelope for any retrovirus. Okay. And it has LTR for, LTR is the number of transcription factors are there which drives this gene. Okay, and then there are signals for integration. It is important, just mention one word, that mouse retrovirus, it has gag ball and envelope, but it does not have these small open reading frames, VIV, VPR, TAT, REV, it does not have these. So here is one example where people can study molecular evolution of how different genes can come into picture. Just to show, yes, we have people get infected, lots of viremia, and then, so early in the infection, you see CCR5 using virus, Later on, you see CXCR4, which is present in T lymphocytes, CCR5 present in Langerhans cells and macrophages. Okay, so I started my work at NII asking the question, is there a remarkable mutation that is, so if in this cartoon, just focus here, you see this virus, macrophage tropic virus, and here is a T tropic virus. Macrophage tropic virus, HIV, which infects macrophages, and T lymphocyte tropic virus, which will help T lymphocytes. Okay, T lymphocytes, it uses another chemokine receptor called CXCR4, and CD4 is common. And this is where the macrophage tropic virus, it uses CCR5 and then CDC4. So if all of them are compatible, you will have this infection. Okay. Now, we asked the question, do we have a remarkable mutation that was reported sometime back? Maybe just 1 year back, actually, when we published this work. Do we have this remarkable mutation in India? Okay. So this is our first paper, and where we precisely had shown, and those days, this paper was in we had this paper ready in 1997. And if you remember the history of this HIV co-receptor, Ed Berger showed it in 1996, late 1996. So we had been very competitive. Okay, so we, and not only that, in this journal, we have traced the inheritance of this mutation. So we precisely mapped this mutation. It was a watershed moment in my lab, with all humility. The moment we read this mutation, said, oh, we have made. So even today, many of my colleagues would call me a Delta 32 lab. Okay, so we found this Delta 32 lab, and a lot of other therapies. We also made some DNA enzyme and catalytic ribozymes against the CCR5, which is all published. So regarding distribution, extremely rare. We found one heterozygous out of screening 100. Okay, initially we screened 15 only. We were lucky we found one, and then we did not find any. So we see heterozygous. Okay, even if we find with great difficulty heterozygous, no homozygous, deletion for Delta 32. White people are 1% homozygous. So genetically, we are at a disadvantage. Okay, so this is where it fits more with the theme of the name of this institute. And then I'm not showing the slide here. We followed it up. There are other, this molecule CCR5, which the virus absolutely needs to get in and establish infection. Okay, this molecule also has other rare mutations. Okay, and we have published later on that yes, there are other mutations also, which is a fairly recent work. I'm not showing the slide. It's all published. So what are the consequences? From a 7 transmembrane, you're down to 4 transmembrane, runs into a termination codon. It's a 32 base pair. Okay, it interferes with the frame, so you run into a termination codon. So what have people done? Okay, can we exploit this? Okay, because 1% white people are homozygous. So this is an ideal situation. Nature has given you an opportunity to interfere, okay, with this. So this was the experiment done. I'm not going to go into any detail other than to tell you that yes, CCR5 Delta 32 homozygous cells were taken out from one person, transplanted into another person. That person was cured of HIV. Nobody talks of cure. That is, so now with the CCR5 molecule, people have started calling cure. Okay. So that was about my work, I thought, from the past. But let me discuss now this work very briefly about this ligase, the CHIP ligase, which we have just published fairly recently, actually. And Amjad, and they have been rather remarkable. One has to be very lucky to get good, dedicated students. So let me show you just once that if you have this chip, okay, MIC-TAT, and if the moment you add MIC-VIP, let's say the TAT has disappeared. So essentially what we are showing is that the CHIP ligase is responsible for degrading TAT. And if you look at this experiment where you have HIV and when you provide CHIP, all these are essentially beautiful blue cells telling you one infected foci. Okay. So if you have this chip, transfect, then you see less number of. So this was published fairly recently, and I would not go into any other detail, okay, about this work. And chip protein leads to stats stabilization, okay. Knockdown. If you knock down chip, then the TAT is not degraded inside the cell. Okay, this is precisely what these figures that they tell you. Then I'm not going to touch much on this. This is what we just published, and NEF actually is responsible for stabilizing P53. Okay, no, I'm sorry. Degradation of NEF. If you just look at this figure, okay, if you go on increasing the NEF, okay, you see more ubiquitination of P53. So NEF is one such protein which is degrading P53. Okay. And then same thing here, and we found out which ligase is important. It is E6AP. So if you have E6AP, you get, for example, here you get more ubiquitination of P53. And then WIF also is responsible. Let me go. So WIF is responsible for here also the chip is responsible for the degradation of WIF, and WIF is known to antagonize apobic 3G, which is responsible for creating mutations in the genome. Okay, so all these are published, so therefore I will not spend much time on this. Oh, this is my very favorite work, which was done by Sachin Verma in my labs some time back. So what we showed that there's a protein called VPU, and VPU actually stabilizes P53. See, if you have this VPU-C means VPU coming from subtype C, it stabilizes P53. Okay, mutant VPU where the beta TRCP motif was removed, it does not do it. So here is a scenario. Okay, this is what we say. NEF is responsible for degradation of P53. If you have more P53 in a T cell, let us say, and if you have, if NEF is degrading, VPU is increasing P53, so there is a balance, okay, and this balance is in favor. If you have more P53, okay, and if you have less P53, it has directly related in the T cells, okay. So we needed to show everything in a relevant cell line, which is the T cell line. Okay. Now this is work. I'm not going to present to you any data, but I was very surprised to see that one such protein alone can interfere with the ubiquitination pathway. Okay, it interferes completely with the cellular ubiquitination pathway. That was very surprising to us. And then we showed that this is actually redirecting the host some host restriction factors towards this ubiquitination pathway. Very excited about this work. First time, as I was telling you, HIV depends on these two proteins, TAT and REV. Okay, TAT is the engine driving for HIV, and REV is responsible for all kinds of gene expression, late gene expression. Okay, so let me show you all. Snehata and Amjad, they were very critically involved with this work. So nothing, all I want to show you is you add REV and the TAT has disappeared. So this was the first case where REV dependent TAT disappearance was shown. And of course, when this is a case of one regulatory protein responsible for regulating another protein, okay? I don't know if there is an example in virology in this kind of observation. That's why, with all humility, we made it to Nature Communication. And we had to do so many experiments to prove our point, okay? We had about 12 to 13 supplementary figures for this paper. Now let me show you some work that the spike protein, what it's doing in presence of COVID. So spike protein, you transfect, and then it gets loaded with these two microRNA. Okay, these two microRNA. If you just look at here, 590 and this 148A, exo is loaded with this exosomes are cargoes for these microRNAs. Okay, what do they do? So here is a paper that we just published that this microRNA which gets loaded, it interacts with here with this 3' UTR of this deubiquitinase USP33, okay, which reduces this level, and this is responsible for degradation of IRF9. So this is how it interferes immunologically. And this is, again, we showed that the case of dengue virus, the same thing, that gets loaded with this microRNA. And then, so this was published in Journal of Immunology. So in nutshell, this is what it is. Again, microRNA 148A gets specifically, which has the target and this 3' RNA of UTR. And then this is this results in the accelerated degradation of ATF3 and uncontrolled inflammation and all that. So this work. And then I'm now coming to my very last slide. So HIV co-receptor has 32 base pair deletion, which we reported in Sajid Hussain was, and those days we were running long gels in the lab, big long gels, okay, and we read that sequence in my lab. So that was the moment which we'll never forget. Then TAT chip NEF is a work of Amjad Ali. VP3 stabilization, now he's in Salk Institute. Then VPR ubiquitination, TAT refs, Neolatana, and dengue virus. I've been fortunate enough to be funded very generously by major funding organizations. And so with that, this is my last slide. And I wish to thank all who are participating and all the faculty, and really thankful for giving me this opportunity to present this work. And Soumitra, once again, a very happy birthday to you, and very, very productive years at Indian Institute of Science, Bangalore. Thank you so much.
Thank you.
Thank you, Professor Banerjee, for your enlightening talk. And there's some time for discussions. If there are questions, Mohua?
I don't see any questions in the chat box yet.
So Professor Das has raised his hand. Maybe he can ask you may ask the question.
Right. So Akhila, it's a wonderful talk. I have been following your work, and you are the pioneer in the molecular virology, the field. I was just wondering, this MID148A in dengue, can you elaborate? So it targets the 3' UTR of the virus or the host?
No, not virus. One of the deubiquitinases, as I showed you. USP33, yeah.
Right.
And
you also had another observation that the dengue and the neuronal cells, they're a crosstalk, and there also you pointed out some microRNA, right?
Correct.
Right. What are those microRNAs?
Okay. So Soumitra, you can understand my difficult position. I think those two microRNAs are common, the 148A and then the other one that we found in dengue as well as with COVID spike. So it was rather remarkable to see that spike itself was able to interfere with all this immune regulation pathway. And those papers are already out. Nitty-gritty, I have to contact Ritu again.
No problem.
Sure. So Parthoda, are you listening? Parthoda is there?
No.
Okay. So before
I'm here.
Okay, Parthoda, you have mentioned at one place Soumitra Das as virologist, or what is it? Yeah, So yeah,
Professor virologist, I said.
Yeah, professor virologist. So now onwards, you have a very difficult task of defending. Then you're the first nation's professor virology. Congratulations, Sumitra. Okay.
All right.
Okay, Shumitro, I take off. I have some other commitment. And once again, thank you so much. All the good wishes, good health.
Thank you.
Yeah. Okay.
Thank you, Professor Banerjee. Thank you so much.
Thank you, sir. Okay.
Thank you, Professor Banerjee, for your excellent presentation and for sparing your time to join us for this.
My pleasure. My pleasure. My pleasure.
So we are ahead of time. We can move on to the next presentation, and that is by Professor Sushanta Roy Choudhury. And Sushanta Roy Choudhury, he did his PhD from the.
Could you please stop sharing, sir?
Oh, yes.
Stop sharing your screen.
Yeah, Professor Banerjee. Are you there, Professor Banerjee? Probably he's not there.
Maybe Vikram, you can push him to the lobby.
Yeah, I have done it. I think if Sushanta starts sharing his screen, then it will.
Oh, then let me finish. Am I audible?
Yeah.
Okay, let me first introduce Professor Sushanta Roy Choudhury. Sushanta did his PhD from the Chittaranjan National Cancer Institute and got his PhD degree from the Calcutta University. Subsequently, he did his postdoctoral work at the University of Pennsylvania, and after returning back to India, he joined the CSIR Indian Institute of Chemical Biology, where he served as the chief scientist and professor of the ACSIR. After his superannuation from IICB, Professor Roy Choudhury joined the Saroj Gupta Cancer Center and Research Institute, Kolkata, as the Chief of Basic Research. Presently, he is at the CSIR IICB as an ICMR Emeritus Professor, where he continues to actively drive his research program. Professor Roy Choudhury's research is focused on the molecular understanding of the genomic instabilities and functional consequences of gain-of-function p53 mutations in human cancer. Besides, his group is studying cancer cell plasticity in ovarian and oral cancers, and they are also trying to develop 3D organoid cell culture model and plasma cell-free DNA-based diagnostic assays in ovarian cancer. He has published well, and he has also been bestowed with the fellowships of all the three national science academies, the NASI, INSA, and the Indian Academy of Sciences, as well as the West Bengal Academy of Science and Technology. And he's also a JC Bose National Fellow. He has also served as the president of the Indian Association of Cancer Research for a period of 3 years, and it is essential that I mention that his association with NIBMG has been long-standing, and he has served on various committees of the Institute, like the Faculty Selection Committee, Scientific Advisory Committee, and Technical and Purchase Committee, etc. With this, I request Professor Sushanta Roy Choudhury to share his slides, and he'll be talking on p53 and microRNA crosstalk in driving cancer cell plasticity. Professor Roy Choudhury, kindly share your slides.
Very good morning to everybody. Sharmila, can you hear me?
Yes.
Okay, let me try to, can you see my slides?
No.
Not yet.
I don't know what. the up arrow, can you see now?
No.
So up and down arrow says the shear tray, right? Open shear.
Yes, shear tray. Yes, shear tray.
Sushantana, yeah, if you can open your PowerPoint slide and keep it maximum, then go to the up arrow and select the window and select that. That will happen very quickly.
When I do that, I see. Yeah. Okay. It worked in the morning, but let me see now. Can you see my slides now?
Yes, it's coming.
Yeah, it's coming.
Yeah.
Is it now? Is it in
Try to move your slide.
No, make it sir, full screen. Huh? Full screen mode is not there.
Yeah, full screen and
is it now full?
No.
No, your slides are not changing.
Yeah.
Now good.
It's working.
Yes.
Yes. Yeah. Okay. Oh no. Why this light is coming? Can you just hold on for a second? Oh no. Just a second. Wrong slides has been opened. Can you see my slides now?
Yes. You have to go to the yeah, fine.
Okay. I think it's now it says 28 January NIB.
Yes.
Okay, thank you very much. Sorry about some confusions. Okay, good morning everybody, and thank you Sharmila for your kind introduction. I have been associated with this particular institute for so long from its beginning. I really cherish a lot of memories with these institutes, and really I am so happy that when Soumitra came, please pardon me that I call him as Soumitra, not Professor Das. When he came to this institute to steer this institute to greater heights, we are very happy and fortunate to have him. With Parthoda, we have been working together to make this unique institute, which is is unique in nature. That it was it's a very unique vision that genomic information at the population level can be utilized for the health and disease of the humankind. That's the mission of this institute, and together with Parthoda, we selected faculties and all these things. We did at the initial time when this institute is trying to blossom. At that time, Parthoda has to go, and then Soumitra took the mantle and carried over to a great extent. I have been very fortunate to closely associate with Soumitra. Although Soumitra and me, so when Soumitra is a PhD student at the IICB, I was not there. But when he returned to India and joined the Institute of Science, we came close to each other. I had some interest in virology also, because I did my postdoc in hepatitis B virus. So he was a pioneer worker at the hepatitis C virus. So we had some interactions. And later on, obviously, as I allude to that, I came to know more detail of Soumitra's another aspect of work, which is his P53 work, which I will just mention a bit. But before that, I really thank Soumitra to allow me to look very closely the scientific activities of the institutes by being a SAC member. And I have seen very closely that how Soumitra is implementing his vision, for to take these institutes to a greater height. Okay, so it was very fortunate for me that I had been the SAC member of this institute and closely monitor their scientific endeavors. With as my work, as you all have been said, that lately I am concentrating in the biology of P53, one of the very important tumor suppressor gene, which has its function is as spread as everything in the under the sky. As you just said the previous lecture, even HIV has something to do with P53. When we talk about P53, mostly we talk about a particular molecular form of P53 of the wild type, but there are other P53 has several different transcripts as well as alternative protein forms, and Soumitra has elegantly pursued that alternative forms of P53 and how those his main fundamental contribution, that how the translational mechanisms, translational function is operating to form various forms of P53. So he one of the delta 40 P53 that he has shown elegantly, that how it is translated by alternative IDs elements and its function as well as its even they have identified some mutations in this which are affecting this synthesis of this particular alternative forms. So I guess because of this immense contribution in this particular P53 biology, I have been asked to say something about our own work in this about this very particular enigmatic protein. So as you can see, my title says P53 and microRNA crosstalk in driving cancer cell plasticity. So, moot point of this of my talk today is the cancer cell plasticity. As you know, the cell plasticity, cellular plasticity, that means that a cell can be present in a different cell functional state. This particular realization, although it was not very initially appreciated at the initial days of the developmental biology, but afterwards, even in the normal cellular physiology, it has been realized, appreciated that a cell can during its differentiation can have, during its journey, can have alternative functional state. Okay. Now, with respect to the cancer cell, cancer, it is now, all our concept about the tumor, the cancer is now getting shattered by realizing that this tumor we cannot see as a monologue. There are various lots of heterogeneity is there among the cancer cells, and one apparent very clearly have been established quite now, quite solidly, that cancer cell, the mutational profile of different cells in a given tumor are not same. There is a mutational heterogeneity, and how those heterogeneities are impacting on the function of those cells that we are trying to understand now. Now, apart from this genetic heterogeneity, also it appears there are some partly because of this genetic heterogeneity and partly because of some other non-mutational event, there is a great degree of cellular plasticity within a given tumor. There are different cells within a given tumor which are functioning differently, and this particular realization that not all cells look alike in a given tumor, has been very enigmatic to the cancer biologists, because whenever we are trying to develop some arsenals against this tumor, we really see that the clever tumor always comes out with some alternatives and shows the resistance. As well as also we have a very poor understanding of how and which cancer cells from a primary tumor actually moves apart, disseminates from the primary tumor and go to another site for the metastasis. Okay, so these two particular obstacles to understand the combating cancer is now getting more information is coming that cancer cell plasticity has a lot of things to do because of these two particular phenotype. And obviously, what I am going to do is in this particular talk is to appreciate you or impress upon you that how the same P53, the mutant P53, I'll be talking mostly of the mutant P53. Soumitra has mostly worked on the wild type P53. I'm going to talk mostly on the mutant P53s and how P53 and the a new era in RNA biology, a host of different non-coding RNAs that is getting appreciated, that they have an immense role in fine-tuning the cellular processes within the cell, how their crosstalks is actually determining in the cellular plasticity. And I will just discuss only one small story that we have recently uncovered in this regard. So as you know, going back to the old days, the Waddingtons, Conrad Waddingtons initially proposed this developmental process of a cell is a kind of a progressive, unidirectional, and irreversible. Okay, but as over the years, it is realized that this particular is looking like a down the hill, a ball rolling down the hill. But it appears now it's becoming apparent that that is not the case. There are instances in normal cellular process as well also various other cellular contexts like injury and others. Thus, the differentiated cells can move back, can go back either by the process of de-differentiation or by process of trans-differentiation through indirect or direct way. This is very apparent in the normal cell physiology now, and that gives the plasticity of the cells in a given tissue. Now, this has become very important in the context of cancer, and realizing that this particular importance of this plasticity, and as well as some other aspects. And I was so fortunate to that I just came after this paper just got published this month, that Hanahan has to revise his original hallmark propositions to add 4 more different hallmarks. That is the one of them is the unlocking phenotypic plasticity, and also as I said that we will be discussing the non-mutational epigenetic reprogramming as one of the enabling characteristics, as well as the senescence, cellular senescence and microbiome. These 4 new hallmarks have come into the picture, and we'll be discussing this thing for now for 10 years to understand that how these new hallmarks are helping the cancer cells to survive in a host. Talking about the phenotypic plasticity, this has been realized that the cells, terminally differentiated cells, also can move back to the progenitor cells or stem-like cells. Okay, that is because of either de-differentiation or maybe some block differentiation. The stem cells cannot differentiate to the mature terminally differentiated cells, but it stops and in some intermediate process, or it can trans-differentiate. That means one type of cells can differentiate to another type of cells. These processes are now happening, and within the cancer, in the tumor milieu, this is also happening. Now we will be beginning to understand that what cellular process, what biochemical process, which genes and pathways are involved in these different processes. Now, obviously, one of the potential mechanisms that we know that in the process of tumorigenesis is the mutations. Okay, we know that the mutations takes place in different genes, and as a result, cells behave in a weird way, and cell proliferates profusely. Okay, but it is becoming also clear that together with this genomic instability, obviously there are some epigenetics like methylations or transcriptomic alterations, as in here it is shown is the alternative splicing, but I will try to impress upon that whole host of non-coding RNAs, especially the microRNAs, are also playing a huge role in this particular process, as well as the post-translational changes. So because of this, all these different mechanisms that Hanahan has to introduce another new hallmarks, enabling hallmarks, that non-mutational epigenetic reprogramming that we'll be trying to uncover over the next decade or so. Already, there are some examples of these genes which play the roles in this particular process, like homeobox genes, SOX genes, other transcriptional regulatory genes like KLF4, MEAT, androgen receptor, and most importantly also the oncogenes and tumor suppressor genes, MEAK and KRAS, P53, RB, and PTEN. They are also playing a very vital role. So as you can see, we again boil down to P53. So obviously, I will discuss that how we have found that P53 is playing a role in the process of cancer cell plasticity, and we'll show that P53 and the MEAK, oncogene, they are collaborating to make these cells awful. So when we talk about the P53, as you know, that my recent interest with respect to the P50 mutant P53 biology is a particular set of mutations. Now, as you know, normally the wild type P53, it is acts as a tumor suppressor, but when the loss of function of this P53 occurs, mutation occurs, tumor suppression goes away, so tumorigenesis takes place. But when we look into the various mutations of the P53 genes, the spectrum of mutations, what we see is that the majority of the mutations are in the missense mutant proteins. Okay, so these missense mutant proteins initially thought that they are sitting idle within the cell, but now for last 2 decades, it is now abundantly clear that this protein is not sitting idle. Actually, they are acting as a gain of function mutation together. In one hand, it loses its transcriptional activity, so showing the loss of function for a phenotype. At the same time, it is gaining a new function, and that new function is actually exacerbating the tumorigenic process, and people are trying to unravel or identify, discover those cellular processes that how this gain of these mutants, they are enhancing the tumorigenic process. And recently in one review article, we summarized the whole thing. I'm not going in detail that mutant P53, actually, this is the few several missense mutant P53. Actually, although they lose their DNA binding property, but they interact with a host of other proteins and activates or repress a large number of reporter genes which are involved in the oncogenic process, or they interact with other proteins and interfere with their functions. As a result, the oncogenic process sets on. And as a result of this, there are almost all the hallmark properties of the cancer cells gets altered. And we are fortunate to identify 3 such pathways that are altered by this mutant P53. One in the replication, the CDC7. The other is in a cellular signaling Eprin B2, and another is the mitotic gene UB2C. So, coming to the cancer cell plasticity, 2 recent papers have shown that mutant P53, this gain of function also can cause the cancer cells, impart the stemness in the cancer cells. One through the EAPTAZ pathway, another directly transcripting, altering the transcription of the Yamanaka factors. As we know that wild type P53 generally is an inhibitor of the stem cells. This doesn't allow stem cells to replicate, and it allows to differentiation and development. Whereas the mutant P53, now it is becoming apparent that the evidence is coming that they can allow that they can cause dedifferentiation, makes the stem cells, and as a result enhance the tumorigenic process. So our interest is to try to understand that to get more insight into this, how these mutants can alter the cellular functional state. Actually, this, our earlier observations we reported by Arindam that when a P53 null cell in a P53 null cell xenograft, when mutant P53 gain of function mutant P53 is expressed, there is a huge tumorigenic acceleration of the tumorigenic process. As well as the Kayum, he in his paper in Cell Death and Differentiation showed that that these mutant-bearing cells, this is a SW40 which has a endogenous P53 mutations. They show the high resistance to the anti-cancer drug like Uracil, and which becomes sensitive when we knock down the efflin signaling. So, both these results shows that these mutant-bearing cell lines, they are highly tumorigenic and they are also chemoresistant. So, one of the mechanisms that we observed there in efflin signaling, but we thought, whether this can also alter the cellular state of the tumor cells, meaning the stem-like property. As you know, as of now, okay, I will come to that later. So what we did, you see, when we made the stable lines, in the stable lines, P53 mutant stable lines, in the null line by transcription of the mutant 53, or in the endogenous, by downregulating the endogenous P53, we show, yeah, if we play with the mutant P53, we can show that the various stemness markers are, getting altered, okay, one way or the other. So, and also at the same time, we showed that by functionally that if these mutants are expressing in the cell, then in the stable line, the CD44, which is one of the surface stem cell marker, gets activated, increased by functional assay, sphere formation assay, as well as by LDH assay, activity assay. We show that because of this mutant, the stemness character of the cell gets altered. They become more stem-like cells. So, as we are talking about the cellular plasticity, as of now we know two cell state in a tumor. One is the non-CSC-like tumor cells, the non-CSC tumor cells that we call, and another is the cancer stem-like cells. Stem-like, there are a lot of controversies are there. Not everybody likes to call it stem cells, but let us put aside that. So there are kind of a binary situation that we see at present. Of course, there are many other intermediates are there, and I am very, very fortunate, and I'm very happy to see the NIBMG, Dr. Sandeep Singh is contributing in this particular area. That he has showed that at least in oral cancer there are 4 different cell states are present in a tumor. So let us confine right now with these two cell state, which is the CSC-like state and the non-CSC-like states. And Eric Lender in his 2011 paper said that they are interconvertible, and probably various epigenetic modification, epigenetical functions are playing a role in this particular process. And we were very interested to understand this role of microRNA in this, whether microRNA has anything to do in this process. As in the literature, there are many reports are coming that microRNAs are actually involved in this process. So we asked the question that whether gain of function mutant 53 that causes the cancer cell stemness, whether it causes through the microRNA or not. So, at the initial days, we did a small RNA deep sequencing experiment, actually, getting impetus from Parthoda, I tried to establish a genomic facility at IICB, and we bought some machines and did some experiments. But unfortunately, I could not continue over there. This is the one small piece data that I could come out of this facility at IICB. We did a small RNA deep sequencing of these NSC cells harboring P53 mutations. And we could show that there were 47 different microRNAs which are differentially expressed between these two cell lines, one without the mutant 53, the other with mutant 53, suggesting that these particular microRNAs are probably directly or indirectly regulated by mutant this gain of function mutant P53. And when we did some validation assay, randomly chosen few microRNAs, and we see one microRNA which is really showing, of course, this validation shows the concordance with the sequencing data, but one microRNA we observed that very highly expressed in the mutant bearing cell lines. And this particular microRNA also reported that could have something to do with the stem cell property of the cancer cells. So we wanted to ask whether this mutant P53, this microRNA gets activated by mutant P53, and indeed we, by various again stable line, transient transcription, endogenous mutant knocking down in the endogenous mutant line, we show that this particular microRNA gets activated in the gain of function mutant P53 bearing cell lines. And we wanted to get some of the the generality of this observation as well as the critical implications, and we indeed see the NCCI60 cell line gain of function mutant P53 bearing cell lines has higher expression of this particular microRNA, as well as, of course, not really very strongly statistically significant, but in lung cancer TCGA data, we indeed see there is a trend of gain of function mutant P53 bearing tumors has higher expression of this particular microRNA. Now that the question that we asked, how this mutant P53, actually reviewer asked us, how they briefly show us the mechanisms, how this mutant P53 is activating this particular microRNA. And we are fortunate that we could see that the promoter of this mutant P53 has the MYC binding sites, and by knocking down the MYC, we show that this microRNA goes down, as well as by cheap assay, we show that this MYC binds to this particular promoter of this microRNA. So we could show mechanistically that how gain of function mutant P53, we already is known that it activates MYC, which in turn actually activates this particular microRNA. So obvious this question, the next comes is that this what is the crosstalk, what is the mechanisms by which this gain of function mutant P53 and this microRNA interactions actually regulates the cancer stemness. Okay, so first we have to establish that this microRNA actually induces the cancer stemness into the cancer cells. And again, using the adherent and sphere culture, we can show that this mutant, this P53, these cells have a higher this level of microRNA, as well as by transient transcription of this microRNA, we show that all markers of the intracellular as well as the outer cell markers gets activated. And by sphere assay as well as by LDH assay, we could show that this microRNA induces the stemness in these particular cells. So also, we have to show the contrast. Other way, we knock down all this this particular microRNA, and we can show that all the cellular markers, the functional assays, sphere formation, LDH assay goes down. So it's conclusively said that this microRNA can modulate impaired the CSC phenotype in the cell. So now the next question was that whether this gain of function mutant P53 alters this CSC phenotype through this microRNA or not. Okay, I just ask you to concentrate on this one. You see, when we transfected the mutant, the markers goes up, but when we knock down the mid, the things goes down, suggesting, same as the functional assay, suggesting that actually this mutant P53 can directly alter this microRNA level, which in turn can alter impart the stemness in the cell. So we have identified another pathway through which these mutants can control or influence or modulate the stemness of the cancer cells. Now, the question is that how this microRNA is altering the stemness property. Okay. Now I introduce at this point a very new protein. With this protein, the Q domain containing protein 2, QDC2, actually discovered in 2007. Okay? And it has now taken center stage. It appears it's a very important protein playing lots of role in the cellular process. This is a 32 kilodalton protein which has a Q domain, and this Q domain means it helps in facilitates the intramolecular and intermolecular ubiquitinations. And people are shown that at present my laboratory at the Saroj Gupta there, Shom Subranath is actively working on that, how this QDC2 is involved in the mitotic processes. It was originally discovered as a degrader of estrogen and progesterone receptor, and most importantly, it has been reported that it acts as a negative regulator of NF-kappa B. So two papers that attracted us when we are trying to understand that how this microRNA is regulating the stemness. We found that this microRNA, it has been reported only that this microRNA targets QDC2, as well as the QDC2 is inhibiting the NF-kappa B signaling. So we wanted to see whether the mutant P53 actually work through activating this MIR actually working through this process or not. And indeed, what we see that in our cellular system, this MIR expression reduces the QDC2 level. And if we in this particular experiment, if you see that MIR expression enhances the CD44 level, the stem marker level, but if we rescue this co-express with QDC2, it goes down. So clearly, this MIR and QDC2 and mutant P53 are probably working together. And again, we try to get the support from the generality as well as for the clinical implications. We see indeed in the CCLE cell database. This microRNA and the QDC2, they are negatively correlated, as well as in the TCAG data are not statistically significant, they are negative correlated. So this MIR expression expressed cells have a reduced CDC20 level, and as a result, and as we see that MIR is activated by gain of function P53, which in turn inhibits the QDC2, and probably that is the reason for this stemness. Well, we also had to show that that gain of function mutated also there is a direct connection in this pathway. So we in the mutant cell line we see the QDC2 level is low, the MIR level is high, QDC2 level low at the RNA level also. And again, we did the rescue experiment to show that in the mutant cells when the QDC2 level is low, but we can rescue that phenotype if we inhibit the microRNA, as well as in the again, in the generality of the observations and the clinical clinical support for this observation, we find there is a negative correlation between P53 and QDC2 expression, as well as both in the CCLE cell database, as well as in the TCGA lung cancer database. So gain of function mutant P53 activates the MIR, which in turn inhibits the QDC2 and gives the cellular character, the stem cell character. So obviously, the next question was obvious that how this happening, and what are the signaling pathways that are playing in the role. As you can see, already you have seen the CDC2 inhibits the NF-kappa B. So our target was NF-kappa B, and we indeed show that. In the mutant cell line, this NF-kappa B is highly nuclear localized. But if we inhibit the MIR, the nuclear localization of the NF-kappa B goes away. And this one is a very interesting experiment. We can show that QD in the non P53 lane, the QDC2 level is high. But in mutant cell level, QDC will be low, and when we inhibit the QDC MIR, and again QDC level goes up. And these are some Western blot to show that yes, what we are talking, that really the the NF kappa B is in the nuclear and the nuclear, and when MIR is inhibited, then it doesn't. So all these things proves that this particular MIR regulates the NF kappa B signaling, which imparts the CSC characters of these cells. And this is my last slide. We are very excited about this particular observation that so far, although we know the p53 mutation is random, in all cancers, and in many cancers, a very high number of tumors are mutated p53. But so far, we have been failed to use this information in some clinically important, meaningful manner. None of the tumors they show some clinical significance, like prognosis or other significance that distinguishes that mutant p53 bearing tumors. Okay, so people are not able to use this information in a clinical meaningful way. But when we wanted to see that if with together the mutations, if we take this MIR expression together, we could show that mutant p53 and high MIR expression cells, both in lung cancer and breast cancer, shows very poor, prognosis, overall survival. So we are very interested to pursue this particular thing, that whether we can use this particular observation with some clinical, importance and translational way. So in summary, as you see in the p53 null cells, there is low p53, low MIR. As a result, QDC level is high, so NF kappa B cannot function. But when in a mutant cells, we know it gets MIR level goes up, which in turn enhances the microRNA expression, which inhibits the QDC2. As a result, NF kappa B goes into the cell, and it activates all the stem cell genes. And hopefully, these informations will carry out. We'll take it further to understand this particular process and other microRNAs, whether how they are involving this particular cell state function in the cell in a non-mutational way. So this is the work of one of the last student of my career, Dishari. I must salute her, because he joined my lab when I was about to retire. So you can understand, as a new PhD student, without having a proper mentor around and the proper lab, she had to complete her PhD thesis, and she did this fantastic work and could publish in a very reputed journal. With these few words and the project, I really thankful to my many collaborators who have helped me over the years, and for these projects also. And these are the funding agencies who also helped us to carry out this work. Thank you very much.
Thank you, Professor Roy Choudhury, for your excellent presentations. We are now open for discussion. Mohua, are there any questions in the chat box?
No, we don't have any questions in the chat box.
Okay. So Professor Das, please.
Right. So, Sushanta, wonderful talk, as always. I was just wondering, the mutant p53 and its affinity towards the promoter of MIR324. Why do you think that the mutant p53 is selectively encouraging the expressions or upregulating the expressions of MIR324? Can you highlight that part?
Well, actually, this particular mutant that we have tested, they do not activate this MIR directly. Okay, we try to see whether this mutant p53 binds to this promoter, but we failed. Okay, so then we looked into the promoter and we found the MIR binding sites. Okay. Okay, and then we realize, because MYC is already known that it gets activated in the mutant cells. Okay, so we then did the experiment to show that actually this MYC actually upregulating this particular microRNA. But at the same time, I must add, we have a world to study. I have shown there are 47 microRNAs that we have found differentially expressed in between two different mutant and null cell lines. So, we should look into those microRNAs, how they are playing a role in this process also.
And those are all activated by the MYC?
No. That we don't know. This is the only one that we know. We have, deciphered that more in a mechanistic way that is, it is regulated by MYC. But other microRNAs, we don't know how they are regulated by, whether they are directly regulated or they are indirectly regulated by mutant p53.
Very interesting.
Anupam has raised his hand. Anupam?
Yes. Very nice talk, a very excellent and elegant talk. I would just simple basic question. As a part of microRNA biologist in cancer, how do you think the microRNA has a specificity? The question, very basic question, asking you as you are working the microRNA. What is the take on you that point?
It's a very difficult question. MicroRNA biology and the regulation of gene expression by microRNAs.
And specificity.
Specificity is there, so there's seed sequences give the specificity. But the problem in microRNA, there are twofold problems. One is that one microRNA can regulate many genes, okay, and a single gene is regulated by multiple. So what people are thinking now, that obviously not everything will happen at the same time, okay, always. So probably it is a context dependent. There could be tissue specificity. Those things are there which actually determines that which microRNA, which gene, and how that will regulate the phenotype. So, we are just trying to, people are trying to understand those things because that's a very, that's fine tuning how it happens by microRNA, as well as now other, non-coding RNAs are also coming into play, like link RNA, circular RNA, all this whole host of RNA world is now needs to be understood. How finer regulations is taking place within a cell for a given phenotype.
Sushantata, can I ask you?
Sure.
Is there a possibility? Can you validate these findings in the oral cancer tissue samples from the Indian population? Is it possible to do it?
It is not possible for me to do it. It's possible for because it's possible for Sandeep to do it, I guess. I don't have any lab anymore, so I can give my knowledge. That's all.
Because we oftentimes generate data from the cell lines which are not representative of the Indian population, and we go on to validate our findings on the TCGA data.
Yeah. That's true. Actually, I have been telling people that, as I was saying, that the 47 microRNA that we have seen, look at these various databases, the TCGA, ICGC, all these databases, and see how those microRNAs are. The last data that I showed, is very important. That this knowledge can be used as in a clinically meaningful way, that we can prognostically classify the patients. Okay.
Yes.
I think that is a very important information, and one should look into it more carefully and try to find out that how these informations can be used in a clinical meaningful way.
So we have some questions in the chat box. So if there is time, we can take one or two from them.
Yes, there is time. You may continue.
Yeah, I think I finished very early.
Yeah, so it's from Arunima Acharya. She is asking that literature suggests that oral stem cells show high CD44 with low CD24. So can stem cells show a varying phenotype with respect to CD44 and CD24 expression in a cancer of a specific histological type?
I think the Sandeep has shown, right, that CD4424, that thing is also added by another LDH. So there are 4 different cell state, right, in the oral cancers, and probably he has also seen in the primary tumors also those kind of cell state. But it's difficult. Yeah, we need to look into we need to look into the primary tumors, whether that's what I'm trying to do in ovarian cancer, to see in the primary tumors what kind of phenotypic cells different cells are present, what we are identifying through cell-based assays.
Then we have another question from Sandeep. He's asking, does this mechanism enhance self-renewal or de-differentiation?
Well, I don't know. Because we have not seen, the one lacuna of this work is that we have shown that NF-kappa B gets nuclear translocated. But what this NF-kappa B is doing at the downstream, okay, we have not shown yet. So I don't know which specific pathway it is activating self-renewal or de-differentiation. So, yeah, we don't know.
Thank you. So yeah, that's all we have in the chat box now.
All right. Thank you, Shushantada. It was a very interactive talk, and we have enjoyed thoroughly. Thank you for sparing your time and joining with us.
Okay, thank you. I have not said yet that happy birthday to Soumitra, my beloved Soumitra friend. Okay, thank you.
Thank you, Shushantada.
Okay, I now hand over to Anushua.
Yes, so we will now take a short break, and we'll meet again at 12 for the next talk.
Yes, Professor Umesh Varshney will be the next speaker, and he will join at 12.
I can join now also if that's what you would like.
Oh, Professor Varshney, you're there.
I'm there, but I'm just kind of quietly working on other things.
As you please.
Whatever you like, I can go ahead with that.
Fine, you can start your talk.
All right, so let's do that. And now this is a different computer, so let me do it again. Is okay? I think that part is working. Let's open the file.
Professor Das, you're fine with this?
Yeah.
Okay.
Is my slide visible?
Yes, your slides are visible. Yeah, thank you.
Right.
But before you start, Professor Varshney, let me introduce you to the audience.
Sure.
Yeah. Professor Varshney did his PhD from the University of Calgary, Canada in 1985, and he got trained, he did his postdoctoral training at the MIT in US. He joined the Indian Institute of Science as a faculty member in 1991 in the Department of Microbiology and Cell Biology, and is a professor since 2002. Professor Varshney is the Jane Tata Chair Professor, J.C. Bose National Fellow, and Dean of Faculty of Science at IISc Bangalore. He is known for his studies on protein synthesis and DNA repair in E. coli and Mycobacterium tuberculosis, and has published high-quality papers in reputed journals. Professor Varshney is an elected fellow of all the 3 National Science Academies of India, and he is also a recipient of the National Bioscience Award for Career Development given by the Department of Biotechnology, Government of India. And he has also been bestowed with the prestigious Shanti Swarup Bhatnagar Prize for Science and Technology in 2001. And he has also received the G.N. Ramchandran Gold Medal for Excellence in Biological Sciences and Technology in 2004. He has held many positions in various committees of different departments of the government, and we, the faculty members of NIBMG, are in fact very lucky to have him on our internal committees like the Faculty Selection Committee, the Promotion Committees, etc. So, and moreover, he's a close associate of Professor Soumitra Das. So with this background, I request Professor Vashni, we are all eager to listen to your talk, which is going to be on UDGX, an unconventional member of the Uracil Dna Glycosylate family. Professor Vashni, please.
Thank you so much for that, very generous introduction. And I have to say that, Soumitra is a great friend, a very good friend of mine. We've been interacting ever since Soumitra joined ISC. I think that was in 1998, maybe. Yeah, he was in the same building that I used to be earlier, Center for Genetic Engineering. Of course, by then we were all part of microbiology and cell biology. So it was the moment Sridhar wrote to me, I think my acceptance to speak on this occasion must have been the fastest. I think within seconds, I think I had responded to that email. Of course, it took me time to get ready with my presentation, but I think, it was really pleasure for me to be at this occasion here. So, Soumitra, very many returns of the happy birthday. It's really a pleasure. And while I think NIBMG people may not be all that happy with you leaving from there, but I'm extremely happy that you're joining us back. So welcome back. It's been going to be, we are waiting for your return for a long time. So I think this will be something very nice that you're going to be back here, and we'll resume our interactions. Soumitra and I, our interactions go a long way. We've, organized meetings together, many of them. We have worked together in many committees, and, Soumitra has always been there to rescue me whenever there has been difficulty. Soumitra has been very kind to rescue me out of those difficulties. So I think, I can, keep going on, but I think, point is well made. That, I'm very happy to be at this occasion here, and I look forward, Soumitra, for your return here. Soumitra has contributed a lot to the institute and the department here, and he will, I'm sure, continue to do so after his return. With those few words, let me talk to you about this work that we've been doing for some time. Now, this is the work that, sort of happened serendipitously. We weren't really looking for it. We were looking for something different. And then we found this protein UDGX. And because it is a talk at NIBMG, and to also sort of fit the format of the symposium title, I thought this will fit better than my routine work on protein synthesis. Towards the end, I will also talk about some of the applications that we have made with this protein, which I think many of you may find useful. So let me tell what this thing is all about. So all of you would be familiar with this phenomena of cytosines in the genome getting deaminated, that is the 4-amino group getting deaminated and getting converted to a keto group. That then leads to conversion of a cytosine to uracil. Now, that happening itself can be a problem for the genome if this particular uracil is not removed prior to the next round of replication. So the cells have a dedicated enzyme, in fact, many mechanisms, many enzymes that carry out this activity. The enzyme is known as Uracil Dna Glycosylate, and the function of this enzyme really is to cleave this glycosidic bond between the base and the sugar. So once this base is removed, the DNA at this point become abasic, and subsequently other enzymes will come in to then reintroduce cytosine at this point. We will not go into that because I'm mostly going to be talking about the mechanism of this bond cleavage and our discovery of this particular enzyme, which does it in a very strange way. So with that, as I mentioned, that there are many proteins that carry out the function of cleavage of the glycosidic bond. These proteins have been classified into 5 different families. And the basis of this classification really is defined by the presence of 2 conserved motifs in these proteins. So motif A and motif B, they are somewhat different in these different families of the protein, although they all actually fold pretty much in the same way. They all are, having pretty much of this alpha helix beta pleated sheet fold kind of a thing, as they call, which is common to all, but they are slightly different in terms of the kind of motifs they have, A and B. And based on that, the substrate specificity also changes. I would like to just kind of, tell you a little bit about this particular protein before I move on to the other, the one that we are interested in right now. This is a major family, family 1, which used to be called as UNG, is actually the major protein, and this is also the enzyme which is very highly active. Other enzymes are not all that active. And one of the other defining properties of this family 1 protein is that this is inhibited by a phage protein, which is called as UGI. So this protein-protein interaction is very tight. You can take up to several molars of urea, and UGI will still remain attached with UNG. And so this is a proteinaceous inhibitor, and many of the bacteriophages actually code for it simply because their genomes have uracil, and they would not like to have a major uracil DNA glycosylase activity to be there in the cell. The other thing is that this family of protein works both on the single-stranded as well as double-stranded DNA. Now, let me just talk to you about this particular protein, which is family 4 protein, because that's what I'm going to get to. There is a slight difference in the motif A and motif B. Sometimes I will end up calling them as motif 1 and motif 2, but they are one and the same thing. This particular class of protein also works on both single-stranded and double-stranded DNA, but it has an iron sulfur cluster protein, 4 iron 4 sulfur cluster protein. This is an iron sulfur cluster protein, and therefore it also has a distinctive color to this. And it is also a thermostable protein, and it is not inhibited by UGI. And the reason I point this one out is that many of the enzymes, when we do it with slightly less purified preparation from the cell, we add UGI to it so that family 1 protein activity is completely inhibited because it's a very tight inhibitor. And we can then just be looking at the activity of the protein which does not bind to UGI. So with that, a small introduction about the different families of uracil DNA glycosylases, let me come to this mechanism that has been defined based on the structure that was solved for family 1 protein. And this is now a fairly old story, roughly the same time that Sumitra joined the institute here. And the motif 1 is defined by this sequence GQDPY, and the other one, motif 2 or motif B, HPSPLS. So what is really happening is that this histidine is actually making a contact with this 2-keto group. And in doing so, it weakens the glycosidic bond here. And the moment the DNA having uracil binds into the enzyme, you can see that there are many other contacts that are being established. And they really define the specificity of this. The enzyme is highly, highly specific. It will bind to nothing else but uracil, and that you can see, because if it was cytosine, it will be an amino group here, and these contacts will not be established. And of course, if this was amino, this will be N, and therefore this contact will also not be established. So it's highly specific for uracil, and this interaction with histidine in motif B is very crucial in cleavage of this glycosidic bond, which is connecting this uracil to the sugar moiety here. And the motif A has a very important role of activating a water molecule here. And this water molecule then makes a nucleophilic attack onto this C1 prime position. And that then makes sure that the uracil goes away. And then you are here having this APDNA, which can then be further repaired by downstream processes, which I'm not discussing right now at this point. So motif A's role is to activate or sort of generate a nucleophile, and motif B actually makes a contact with uracil, which weakens this glycosidic bond. Now, if we come to Mycobacteria, this is where we discovered this protein. Mycobacteria are very GC-rich organisms, and so the chances of cytosine deamination would also be quite high. And when we looked at the genome, we found that there is a family 1 protein, UNG, which is similar to that of E. coli. And then we find that there is a family 5 protein, which we named as Udgb. Now, given that this is a very GC-rich organism, and it also actually goes into macrophages where it is subjected to acidic conditions, so there is a lot of reactive nitrogen and reactive oxygen, one would think that there would be a lot more cytosine deamination to uracil, and then just having these 2 uracil DNA glycosylases was somewhat puzzling to us. And so one of the postdocs in the lab, Srinath, he started looking at the genome by doing a search for family 4 protein. And he did actually find a protein. So this is a family 4 UDG, which are already been characterized in many of these bacteria. And what he found was that there is a protein which is not exactly family 4, but similar to family 4. You can see that so much of homology. But then in case of the UDGX protein that we named afterwards, there is a very distinctive sequence here, KRRIHK, which is not found in any of the family 4 proteins. And so this was interesting. And so he actually mentored one of the integrated PhD students here, who then cloned this gene from many organisms, including Mycobacterium smegmatis. In fact, it turns out that all of the organisms which have UDGX, and we know many more now, they're all GC-rich organisms. So there was a connection that this particular protein is found in GC-rich organisms. So the idea then was that he should clone this protein and look at the activities. So he cloned it, and he was able to purify it to a very good quality. The very first thing that he observed was that the protein was brown in color. And that's a good indication that there is some iron sulfur cluster in this particular protein. Basically, it absorbs around 420 nanometer. And so he then wanted to look at the activity of this protein. So he obviously did this assay where we make use of a DNA which has uracil. So this is a synthetic DNA, and it has uracil in it. Now, if the uracil is actually removed by the glycosylase activity, there would be an AP site that would be created. And if the AP site is treated under alkaline conditions, this DNA would actually be cleaved into 2 parts. Now, one of this part, which is 5' end labeled with P32, would show up as a shorter DNA compared to this one. Unreacted will be this size, and the shorter will be only this size. And we can see that. So here is the DNA oligomer, which has not been reacted by any protein with any protein. And you see that much of the DNA is right where it should be. When we treat it with the family 1 protein, which we've been working for a long time, you see that much of the substrate has gone into a product which is migrating faster, as you would expect on this particular gel. But when we now treat this DNA with UDGX, which he purified over here, rather than seeing a fast migrating band, he started seeing a band which was sitting right into the well of the gel. It migrated very poorly into it. And it sort of gave an indication that this is something which is making a complex with the DNA. Now, the interesting part was that he took this complex and treated with proteinase K. And what you see here is that it is still migrating slightly above the substrate, indicating that there is actually some peptide attached to this particular DNA. And so that became quite interesting. It sort of indicated already, because, you know, if you ask me what is in this gel, there is actually 8 molar urea in this gel. And before we load this protein onto this gel, it is heated in 8 molar urea with formamide, dye, and all that kind of a thing. And if something is still sustaining this, it's a pretty good indication that it is a covalent bond there. So then what Sang did was he actually used many other DNA with uracil in it. And what he found was that the DNA, which had uracil slightly inside from either the 5' or the 3' end, they actually made a complex. Now, this is a Kumasi blue gel. So this is a protein where no DNA has been incubated. Now, you see, when we incubate it with uracil containing DNA, both of these, the protein has actually been shifted. And this is STS page. So the only way it can do this, that it must be a covalent bond here. So then we did the mass spectrometry. And so if we do the mass spectrometry of the protein without DNA, you have certain peaks. Then if you have DNA, you find a unique peak here. And if you now subject this guy to look at this, M by Z value on mass spectrometry, you find a mass of 2,325.69, which actually corresponds to the mass of the DNA that has been taken here, minus uracil, because the glycosidic bond is cleaved, and a peptide which will arise because we are treating with RC proteinase, endoproteinase RC, and this peptide and this mass together adds up quite nicely to 2325.69 Daltons. So now this experiment showed that the DNA is actually bound to this peptide of the protein, but it still doesn't tell us which amino acid is involved in making the contact with the DNA. Now for that, we had to do ETD, and this was done in collaboration with Eugen, who was a visiting scientist at JNC, and I do have some connection with JNC, so he was visiting me here, and we talked about this work, and he was quite excited about it. So this work was actually done in collaboration with him. Now what we can do is that if you have this peptide, you can cleave it at different positions depending on the energy that it is being bombarded with. If we do the cleavage at this amide linkage here, then you can get two kinds of ions. One will be defined as C, the other one on Z. So we can actually find out so if we were to look at whether this position is involved, we can take the mass of this and we can take the mass of it. And if we subtract this from this, let's say this mass from this mass, Z ion, then we will know which amino acid is involved. And this is the same approach that was used. So now here is the experiment. So this was the peptide to which we know that the DNA was attached. Now we subtract, let's say, C3, look, this particular C3 from C2, we will get this histidine. And that should actually correspond to the mass of histidine and also the uracil minus this DNA oligomer that we had used. And the mass of that is 1469.1, and that precisely matches. If you subtract C3 minus C2, if you subtract this from this, then we get this. We can do the other way also. We can subtract from the Z ions, and again we get a mass of 1469.1. So this really adds up to the mass of histidine and the uracil minus a DNA oligomer that was used. And these peaks are actually shown here, but I will not go into that. But suffice to say that this then identified that the histidine is involved in making a covalent bond. And I should say that covalent we have not yet proved, but all of this really directs us in that direction, and I will then come back to that. So then what Sang had done was, if we look at these motifs, so I had already defined these motif A and motif B, and based on the sequence characterization, he also already knew that the protein that he's dealing with has this as a special sequence, KRRIH. And so we have three very distinct motifs here. Now, if we try to compare these motifs, we can make mutations here. I will come to the mutations of motif A and motif B a little later, but let's talk about the mutations here in this very, very distinctive sequence that is found in UDGX only. If we now mutate this histidine, this is the one which we found out from mass spectrometry is attached with the oligomer. If we mutate this histidine to serine, and this histidine is called as histidine 109, as soon as we change that histidine to serine, you see, now we don't see that complex anymore. We see a fast migrating product. So essentially, we have converted this enzyme, which was originally making a covalent bond, into an enzyme which is now able to do turnover. And we can change it to other amino acids, which are shown right here. In all of these cases, I don't know whether I can get rid of this. In all of these cases, the protein has turned into something which is now giving us turnover, and earlier it was just sitting as a complex. If we mutate 4 of these amino acids simultaneously, then the protein becomes actually inactive, as you can see over here. So this is the DNA oligomer substrate that we started out, and this is after, this is the oligomer that we started out with, and this is after we add this particular mutant to this. And, you see some product here, but that's because, these proteins were not purified to high quality. But when we add UGI to it, you see that there's no product here. And so this protein has become inactive. And likewise, if we change this, arginine 107, which is this guy over here, and you will see that these are actually important residues. Again, the protein becomes inactive. He has then also tried to make changes in the motif A. In fact, I think this data also shows that if you do too much tempering here, the protein will become sort of inactive. As you can see here, F1. This protein has become inactive because we don't see a complex, nor do we see a product over here. But if we just change a single amino acid, that is Q to A, the protein still remains active, and this actually forms a complex. Now, what I can tell you well ahead of time that this glutamate that we have here, E, this residue number is 52. This is also important in catalysis. So as soon as, in fact, we have changed this glutamate to glutamine here, the protein has become inactive here. So then Eugen is actually a structure biologist. He took it to Korea, and he solved the structure of this protein. And this is really, again, the alpha-beta fold protein. And he also did one more thing, that he overlapped this structure of UDGX with the family 4 UDG. And the only thing that you find, it's overlapping pretty well, actually. The only thing that you find is that this particular loop, which is distinct in UDGX, and this is what we saw even in the comparison of the amino acid sequence, that KRRIH loop this was distinct, and it turns out to be very, very distinct. And it also turns out that this is a major thing in the catalysis of the glycosidic bond cleavage. Now here, what I'm showing you is the structure of the family 1 protein, UNG, and the way uracil is taken into the active site pocket. So this pointer now is the active site pocket, and particularly this yellow kind of color that you're seeing, this is actually the active site pocket of the enzyme. But originally uracil is sitting in the stack of this double-stranded DNA. So uracil has to be taken out of this stock and pushed into the active site pocket. And this job is done by the motif B. And motif B, the leucine residue particularly, so HPSPLS, so this L is really the one which you see here is getting into the double helix and pushing uracil into the active site pocket so that the glycosidic bond can be cleaved into the by the residues in the active site pocket. Now, a similar mechanism is actually used by UDGX also, but in a very different way. And so this arginine 184, which is a part of this motif B, is actually getting into the DNA stack, and you see that uracil has been pushed into the active site pocket. Now, there is a histidine 178, which is again part of the motif B, which is carrying out the same job of actually making contacts with O2 of uracil so that the glycosidic bond can be weakened. And this glutamate 52, about which I talked already, that if we change this glutamate to glutamine, the enzyme becomes inactive. And I'll come back to it a little bit more to show what exactly the role of this glutamate 52 is. So in terms of the mechanistic part of how uracil is taken into the active site pocket, that remains, the mechanism remains conserved, except that here we are using arginine 184 as opposed to histidine. The role of histidine 172 is same as this particular histidine. I mean, the HPSPLS, motif B, the histidine of motif B of family 1 protein. And we will come back to the role of many of these other residues in the coming slides. So here is what happens. So this is the arginine 184, which I said is doing the job of leucine in family 1 protein. And you see that as soon as the DNA substrate bind, this leucine, which was originally in this conformation, swings into the pocket to push the uracil or uridine into the active site pocket. And you can see this one again in this view, which really shows you that this arginine is swinging into the action over there. We can now talk about this histidine 109. This is the histidine which is found in that signature loop or the extra loop or the distinctive loop that we had identified, part of that KRRIH sequence. And this histidine, as you can see, is also moving into it. And this, of course, now the uracil is cleaved here, but it is making contacts with this sugar moiety here after the uracil has been cleaved. And I'm going to talk a little bit more about it because this is something very unique about this particular protein. And what shown here is the electron density between the sugar that is in the DNA after the uracil has been cleaved and the histidine 109. And if you look at the spacing between these two, it's about 1.5 angstrom. And this actually quite clearly, of course, the mass spectrometry and the mobility on SDS page already sort of told us that it's a covalent bond. But here is the sort of, visibly we can see that it is actually a covalent bond. Now, if we compare the active site pocket of family 4 protein and the UDGX, remember this is how the student in my lab started out because they were trying to search based on the family 4 protein. The active site pocket residues, they actually align very well. And there is a glutamate in family 4 protein, and that is represented by glutamate 52 in UDGX. The major difference that we find here is that in family 4 protein, there is an asparagine, and that asparagine actually does the job of fixing a water molecule. And this is really essentially the same mechanism that is there in family 1 protein, that this water molecule, activated water molecule, will make a nucleophilic attack on the C1 prime of the sugar and will, of course, cleave the bond. Uracil will leave. But here, when we have histidine in place of this asparagine, there is no room for water molecule to come. Instead, this histidine is making a direct nucleophilic attack onto the C1 prime position. And this is how actually we end up making the covalent bond with histidine. Now, I mentioned to you that glutamate 52, which when we mutated to glutamine, was making the enzyme inactive. And that point is also very clear, because what it is doing is actually it is making the histidine 109 a good nucleophile, because it's taking away this proton from here. So this mechanism turns out to be quite good. Now, let me talk to you about this serine. When we had mutated this histidine 109 to serine and many other residues, it then started giving us a turnover. Now what happens is, this is also based on the structure. We now see a water molecule coming to, I'm sorry about this block that is actually blocking this thing, but I'm sure you can see this water molecule here. And this then becomes the same mechanism as we saw here. So this water molecule can now provide turnover to the enzyme. So rather than histidine 1, now we have serine and this water molecule, which will now be, making a nucleophilic attack. So then Shashank, who joined the lab, and he's done many of the other experiments also, he made a whole bunch of mutations, essentially validating. So he's made mutations at arginine 184, he's made mutations at histidine 178, which both are part of this motif B, he's made mutations at glutamate 52. So all of these mutations he's made in combination as well, and they're all as per the expectation. As soon as you mutate this one, the activity goes. You mutate this, activity goes. You mutate this, activity goes. So that is quite well validated. So that then allows us to propose a mechanism which is essentially based on the family 4, except that we bring in histidine 109. So here in family 4 protein, as I mentioned, that there is this asparagine which is activating the water molecule, and that can actually make a nucleophilic attack here. This histidine doing the same job of interacting with O2 of uracil, which weakens this glycosidic bond, and then you go through this intermediates to give rise to a free uracil and of course this APDNA. But in this case, what is happening is that there's no water molecule. The nucleophilic attack is being made by this histidine 109, and as soon as that happens, essentially what you've done is you've linked this protein to DNA, because this now becomes a covalent bond, as you can see it over here, and you have this protein DNA complex. Of course, uracil has been released. So it is still a uracil DNA glycosylase because it's cleaving the glycosidic bond, except that it has trapped the DNA into a covalent complex. Now, to further confirm the mechanism, as I mentioned to you, Shashank made this mutation of E52, and the idea is that it is actually enhancing the nucleophilic attack, quality of histidine 109. And as soon as you make that mutation, E52 to glutamate, now he's done a little bit more, long-term reactions. You will see that this is the wild type protein, and you start seeing complex in sort of 20 seconds. But here, in 20 seconds, you see very little complex because histidine 109 is still there. And if you keep it for quite some time, you will see some complex, but nothing compared to what you see in case of wild type protein. And if you change this E52 to alanine, it becomes even worse, as you can see it over here. Now, the other thing that we wanted to really show that this histidine is the one that is really making a nucleophilic attack. And what we then wondered was that if we actually lower the pH of the reaction, to something that will be below the pKa value of histidine, which is around, I think, 6.6 or in that neighborhood. What you see here is that then it will not be able to work it will not make a covalent bond here. And what Shashank noticed was that if we do the reaction at 4.5, you do indeed see some amount of product. So here is the control, where 109S mutant, which we know gets turnover, is making the product here. And here is the protein, wild type protein, at a pH of 8.59, which we know makes a complex. You hardly see product here. But at 4.5 pH, you see much more product. So this element of the mechanism that histidine is making a covalent bond or is making a nucleophilic attack on the C1 prime position is also quite nicely validated here. So then what exactly is happening? UDGX is making a covalent bond. That's really not providing a solution, because it's made an adduct with the DNA, which itself is a problem. While it removed uracil, fine, but then the entire protein is sitting on DNA, which is not fine. So in the lab, Indu, who is a postdoctoral fellow now, for her PhD project, she worked on this protein UDGB, which is, as I mentioned to you, is a family 5 protein in Mycobacteria. So what we had noticed was that UDGB also forms a stable complex with DNA, but this is not as stable as UDGX, and it does not form a covalent bond. So then she was interested in understanding the mechanism of how UDGB is removed from DNA. And what she found was that two proteins, UVRB, which is a protein involved in nucleotide excision repair, and UVRD2. In Mycobacteria, there are two UVRD proteins, UVRD1 and UVRD2. Both of these proteins actually, they serve as helicase, and they can remove UDGB from the DNA. And then, of course, it can be reacted upon by nucleotide excision repair, because AP sites are also substrate for nucleotide excision repair, and it can go on for further repair. Of course, the AP site can also be repaired by AP endonuclease followed by REG-J protein followed by the DNA polymerase. But when Sang looked at this mechanism, UDGX was not a substrate for nucleotide excision repair pathway. What he instead found was, and I'm just going to show you one of these data. He's done a lot of genetic experiments where he found that there was synthetic lethality of having UDGX and not having any of these proteins, which meant that there is a genetic interaction between these proteins. And all of these proteins are actually involved in SOS repair or homologous recombination. So he then suggested that the repair of this UDGX complex on DNA must involve REG-A. And here I'm just showing you one piece of data. And for other proteins, it's actually the same kind of experiment, excuse me, except that we have used different mutants. So here, if you notice that UDGX has been used, and to make the UDGX do more of repair, we have overproduced in the bacteria UGI. And as I mentioned to you, UGI will inactivate family 1 protein, which is the most efficient enzyme. And so the DNA will have more uracil, which means more of UDGX will bind to DNA. So if you look at the wild type strain, which grows quite well actually, when the wild type has this complex or which construct which has UDGX and UGI together. But now if we take delta REG-A, you see this guy is very, very sensitive to H2O2 or even to regular growth. And so this kind of interactions then told us that that UDGX must be repaired by REG-A dependent pathways. So based on that, and based on the data that I have not shown you in the interest of time, what we propose the mechanism is that if UDGX finds uracil in DNA, it would make it complex. Now, as soon as the DNA is being replicated, the replication machinery will encounter this complex, which it can't get rid of. Of course, the RACA will be binding to the single-stranded region here. It can then be channeled either to SOS mediator repair or homologous recombination to then resolve this particular region to clear the complex. This is rather, very difficult way of resolving this protein DNA complex, and we're still looking for whether there are any other mechanisms that will remove UDGX from this DNA complex. In the meantime, another graduate student, Madhurima, who has just now finished her PhD, she thought of a mechanism that, if you're making such a tight and covalent complex of UDGX with DNA, can we use it as a probe to detect uracil or occurrence of uracil in DNA? So what she did was she actually made a chimeric construct of M cherry with UDGX, and she also took a UDGX mutant, which is called a C275, and about which I have not talked to you. But what this mutation does is that it does not allow iron sulfur cluster to form and therefore, or to bind to the protein, and therefore the protein actually becomes inactive. So this is her negative control. And what she shows is, of course, when you overproduce this protein because of M cherry, you see this purple color. And the idea is that if this UDGX is binding, it should remain attached. And because of this purple color, it should shine purple. So she looked at the activity. The chimeric protein gives good activity. Her gels are not as good as, of course, Sang had, but nonetheless, these are all complexes. And so this is the cartoon that she has shown, that if you have uracil in the DNA, UDGX will bind, and then we can detect, of course, M cherry by microscopy. She had then done the real experiment where she wanted to look at whether this protein will allow us identification of uracil residue in the chromosomal DNA. And so she's taken bacteria, which is TG1, a regular strain that most of us use, which is UNG plus. So it has a lot of uracil DNA glycosylase. Let's not worry about this DUT at this point. So this, because uracil DNA glycosylase is active, will not have much uracil in the DNA, and therefore one would not see much of this red or the purple color. So in this one, you don't detect any dots which are sort of purple. But if you take RG1032, which is UNG minus bacteria, you see that this protein is now binding to DNA. And all of this can then be also another control she can take, that in this particular strain, if she uses the chimera where UDGX has been inactivated, again, she doesn't see any of these purple color dots. And she's quantified all of this. This is based on confocal analysis. So she sees a lot of protein binding to the DNA none of the hardly anything binding into the DNA in TG1. But when she takes uracil containing DNA, quite a bit of it is binding. And when she takes the mutant protein, again, much of it is not binding to the DNA. So this really then provides us a tool to look at the uracil residues in the DNA. So now let me just summarize it. Essentially, what I have shown you is that UDGX protein will form a complex. We are still searching for a biological significance of it. Of course, we know now that this protein is actually overexpressed, or I shouldn't use the word overexpressed. It is expressed much better, or its expression is induced with the stress conditions like high salt, pH, low pH, and many other, treating with hydrogen peroxide. All of those conditions leads to induction of UDGX. And so we are now actually getting somewhere. Initially, we weren't able to detect those things. Then, of course, we have shown that it forms a covalent bond. H109S is an enzyme which provides turnover. So essentially, we have evolved this protein back to something which will give us turnover, and the repair pathway actually feeds into REC-mediated repair. And we've shown that it's an excellent tool for detection of uracil sites in DNA. I'm going to skip this one because I'm pretty sure I'm short of the time, and basically just acknowledge the people who have done the work. So Pao Biaksang is the one who actually discovered this protein. Uchan is a student in Eugen's lab in Korea who did the crystallography work. Shashank is doing a lot of biochemical work. Madhurima has made use of this protein for detecting uracil sites in DNA, and she collaborated with Kapudeep Karmakar in Deepshika Chakravarti's lab. And Chandana and Shivji, they actually worked on a bradyrhizobium protein, which is again a GC-rich organism. And the interesting part about this protein is that it comes along with additional domain at the N-terminal. And that seems to give it some turnover. It still forms a complex, but there seems to be some turnover, particularly views slightly changed buffer conditions. So that is something for another day. But the work has actually been done in collaboration with Eugen Wu, as I mentioned, Deepshika Chakravarti. Somnath Datta, I did not show his work, but that was the work where we showed that the protein binds to uracil by making use of electron microscopy. And again, Ravi Tiwari's work, collaboration with him, I've not shown you the data for it. We are supported by ISC, DST, DBT, CSIR, and JN Tata, Foundation for the work in the lab. With that, let me thank you all for your, patient hearing, and I'll be happy to take any questions.
Yeah, thank you, Professor Varshney, for your very interesting and wonderful talk. And Mohua?
Yeah, we don't have any
questions.
But as we start the discussion, maybe questions will start flowing in the chat box.
Okay. So one of the things that we still need to work really is what exactly it is doing for bacteria. There are, of course, we have hypothesis. Hypothesis is that, when you have uracils on both these strands, and if you remove uracil, and if you don't carry out the downstream processing, the DNA will be fragmented. And this may be one situation where you don't want to fragment the DNA because it will actually be very, very cytotoxic. That you bind the protein so the DNA remain intact, and when you have sufficient of the downstream enzymes, that this protein can then be, taken care of. So that's one of the things that we are following up, but only time will tell how far we go with this particular protein. In the meantime, many other people are making use of this protein for many of the applications, particularly uracils, finding out the locations of uracils in DNA.
So, Umesh, what is this role of this Uracil Dna Glycosylate in other pathogenic bacteria? Do you think that it can be correlates with the virulence?
I think it might, in terms of, so in case of Mycobacteria, we can correlate it quite well. But the strange thing is, if we want to correlate with virulence, Mycobacterium tuberculosis doesn't have this protein. So something directly relating to virulence in Mycobacteria seems a little difficult to do. But in Rhizobium, bradyrhizobium, you see the bacteria, when they go for nodulation, they have to actually sort of overcome the host defense to begin with. Of course, subsequently they establish, symbiotic relationship. But to begin with, I'm sure host is not going to be very happy to let some bacteria come and invade it. So under those conditions, I suspect maybe this bacteria is going to be helping, this Udgb is going to be helping bacteria. But, that's one example that we can think of. We have also found that in case of Caulobacter, this protein is there. And Caulobacter is another system that we've been working with, because Caulobacter will allow us, monitoring or following this protein through the life cycle. And Caulobacter, as you know, has a very interesting life cycle. And we hope to find out something more by looking at this particular protein in Caulobacter. So, Samitra, I'm not able to answer your question even as of now, actually.
Right.
We really
I was just trying to see whether the levels will, somehow vary, or their status will vary with the progression. That's
right. That's right. Already we know that if we take, let's say, we put smegmatis in 1 mole of salt, this protein is induced. We put it in hydrogen peroxide, the protein is induced. And we actually treat the UDGX minus bacteria with hydrogen peroxide, it is susceptible to it. So I think there is a role for this protein, but we have just not really nailed it down as to what precisely it is doing.
Right.
Are there any more questions?
One question from Dr. Mohapatro. He's asking, Uracil Dna Glycosylate family 1 is seen in Klebsiella.
It is seen in most of the bacteria. It's a very, very conserved protein. Of course, there are bacteria, for example, I mentioned Caulobacter doesn't have family 1 protein. I think Drosophila also doesn't have that protein. So there are now examples that we are aware of where family 1 protein is absent, but by and large, family 1 proteins are very, very conserved and they're found everywhere.
So if there are no more questions, we'll thank Professor Varshney one more time for his excellent presentation. Yeah, we got to know a lot about the bacteria and the E. coli and all those things, which is a very new, I mean, it's very interesting. So thank you. Hope to see you, meet you again in future.
And Samitra, look forward to your return. You're now coming back in the next few days. So look forward to your arrival here.
Right. Thank you.
Thank you. Yeah. Bye-bye.
Bye. So our next speaker is Dr. Sridhar Chinnaswamy, and I'm happy to introduce you as he's one of my colleagues, and he's one of the first to join as a faculty member in this institute. When Professor Majumdar did the recruitments, he joined, and subsequently Dr. Bhashwati Pandit. The two of them were the first to join the institute. So, and after that, Sridhar has developed a nice niche of his work, and he is going to share that with us. He is an associate professor and is an intermediate fellow of the Wellcome Trust DBT India Alliance. Sridhar works on understanding the genetics and biology of type 3 interferons. He is a veterinarian by training, but he has opted to move out of this field, and he has undertaken research in basic sciences. He obtained his PhD from the Texas A&M University at College Station, and he working on the biochemical characterization of hepatitis C virus, or DRP. So subsequently, he moved to India and took up this position at an IBMG in 2010. And since then, he has concentrated his efforts on type 3 interferons and the relevance in human health and diseases, including infections and inflammatory diseases. So Sridhar is going to share his newest results that his group has obtained on the immunomodulatory properties of interferon lambda-3 and lambda-3. Sridhar, you may share your slides.
Am I audible?
Yes, you are.
Thank you, Professor Sengupta, for this kind introduction. And let me share my slides here. Are you able to see my first slide?
Yes, you'll have to do the slideshow.
Yeah. Did it become full view now?
Yeah, full view.
And changing?
Yes.
Okay, great. Thank you again. I'd like to thank the organizers for giving me this opportunity in the midst of such eminent people to be speaking in this symposium. But before starting my scientific talk, I'd like to take this opportunity to say a few words about our beloved director, sir, Professor Soumitra Das. So I know him from actually, first time I saw him was in 2001 when I was doing my master's thesis in Bangalore. So my institute was closer to IISc. So once I went to see him borrow some cells actually in 2001. So interestingly, he remembered that. So I was so surprised when he joined NIBMG somewhere in the first week when I met him. I told him, do you remember that I had visited your lab? And I was so surprised that he remembered, so vividly that I had seen him at that time. So that was really surprising to me. And then I made sure that I never presumed that he would have forgotten anything in the next few years that he was here, and it was great to be working with him. And the other thing that I would like to share is so when it was announced that he would be coming here, so our people here, mainly the admin and the finance people, they somehow got to know that I knew him before, so they wanted to know how he was. So I kind of said that if nothing, he will never ever get angry at you. So he will never put a stern face at you. And he has his own soft ways of getting things done, getting things worked from his colleagues as well as subordinates. So I'm sure every one of them will agree to that now after spending 3 years or more with him. It's really sad that we are going to miss him. But thank you, Professor Das, for everything, all the good that you have brought to NIBMG. I wish you all the best in the future for a great, fantastic life. Thank you. And let me start my talk here. So I will be talking about, I'll not be talking about like the previous speakers who presented work from several years, but I will just be concentrating more specifically in the work that I've done in the last 2 to 3 years at NIBMG. So I'll be talking about the molecular phenotypes induced by interferon lambda 3 and interferon lambda 4 in innate immune cells. So let me begin by introducing you to interferons. Interferons are of 3 classes, as many of you would know. The type 1 interferons are the more ubiquitous and abundantly expressed interferons. They are essentially expressed by all nucleated cells, and most cells respond to them as well. The type, let me make my pointer here. The type 2 interferon represented by the sole member interferon gamma is expressed solely by immune cells, but many immune cells respond to them. And type 3 interferons, the class of interest that the interferons that I am interested in, are the newest discovery. So they were only identified in 2003 after the Human Genome Project. And the newest member in them, interferon lambda 4, is as recent as 2013. It was discovered in 2013. So it was this locus came to the limelight in immunology, so to say, after the famous genome-wide association studies carried out in hepatitis C virus infections. And since then, it has piqued the interest of a lot of immunologists and geneticists. And now we have enough data to say that these type 3 interferons are the frontline guardians of the epithelium. Summarized in this figure by Evangelos Andriokos. If you see the respiratory epithelium, when you get a viral infection, the first line of defense is by the type 3 interferons, which happens to be milder on the host tissue. So if you can get rid of the infection by type 3 interferons alone, which are strongly antiviral, then no harm caused to the host. But if for some reason, if that fails, then the type 1 interferons kick in with a more systemic and more stronger inflammatory activity that obviously will lead to more and more damage to the host. So that underlies the importance, the subtle roles that type 3 interferons are playing in specifically as antiviral molecules, and probably in immunomodulation. That is what I'll show you in my data. They are not just antivirals, but they could be having important roles in modulating the phenotypes and functions of other immune cells than those that are producing them. Okay, so interferon lambda locus in humans is on chromosome 19. It comprises of essentially 4 genes, 1, 2, 3, and 4. And this locus is not only the type 3 interferons are not only interesting recently for their biology, but also they are very important evolutionarily. There are several genetic variants that are identified here, but most of the studies point towards functions associated with these genetic variants, specifically to the expression of lambda 3 and lambda 4. I will get into that in a bit. But as I said, it is evolutionarily important. For example, this figure shows that interferon lambda 3, which was called IL-28B then in 2011, it shows the nucleotide diversity varies across populations. For example, in the African population, as expected, the diversity is maximum, but it decreases in the European and very low in the Asian. The Asian here is basically excluding the subcontinental Asian populations, of course. And so, in fact, the study concludes that the positive selection has targeted these type 3 interferons in non-African populations. Now, interferon lambda 4, the newest member, as this diagram shows, the purifying selection seems to be operating, and that shows the conservation of this protein across different species, maybe except the squirrel monkey here. And that shows that it's functionally very significant. But another interesting thing is that only in humans, a polymorphism, a dinucleotide variant actually regulates its expression. So there is a pseudogenizing allele, which is known as the TT allele. So people having this TT allele will not express a fully functional interferon lambda 4. And it seems that this pseudogenizing allele has been positively selected in the East Asian population. So therefore, interferon lambda 4 has played an important role during human evolution, probably reflecting how humans have adapted to changing environments since the out of Africa migration. So as I said, after the HCV GWAS studies, a lot of people got interested in this area, and people started to test, in genetic candidate gene association studies, and a number of diseases have been associated with the genetic variants here. For example, of course, there are HCV, and then respiratory and GI infections in African children, and malaria again in African children, and also inflammatory diseases like non-alcoholic fatty liver disease, and also cancer, and other viruses like Andes virus and HIV, etc. So I have a table here which shows more diseases that have been reported to be associated with these variants like COPD, Hashimoto's thyroiditis, lupus, which are autoimmune diseases. And also these variants play a role in responses to vaccines against influenza. And also not only in disease phenotypes, but you also see in health phenotypes. For example, this study shows that postpartum women have different ISG expression that correlates with the genetic variation at the interferon lambda 3 genetic variants. And not surprising, studies have started to show up on COVID-19 and its association with interferon lambda variants. But none of these studies actually differentiate between whether the lambda 3 variants or the lambda 4 variants. The problem is summarized in this figure. So they are physically, of course, close, and a phenomenon called linkage disequilibrium is high in most of the populations except the African population, that prevents use of statistical methods to exactly say either these variants or this variant is responsible for the phenotype. As far as the variants are concerned, there are two genetic variants that are known to have functions that regulate interferon lambda 3 expression, and the lambda 4 variant, I told you, the TT allele dinucleotide variant, that either abolishes the open reading frame or creates the open reading frame. So some variants control lambda 4, some others control lambda 3. But in a genetic association study, it is very difficult to differentiate which of them is responsible for the phenotype because of the phenomenon of linkage disequilibrium. So if you get African populations with a disease phenotype, you could answer that question, because in African populations the LD is very low. So another problem is that lambda 3 and lambda 4 are only approximately 30% identical. So they could be having different functions that we need to identify. So using the African populations, a few studies have come up that showed that interferon lambda 4 and not lambda 3 is behind several viral infections and also malaria. And there is what is called as the interferon lambda 4 HCV and now interferon lambda 4 virus paradox, because it is a strong antiviral cytokine. In cell culture, if you add this lambda 4, it will inhibit probably all viruses that can grow in cell culture. But in vivo, paradoxically, it has a risk for example, HCV infections or childhood respiratory or GI tract viral infections and even malaria now reported. So this is called as the paradox. So that has to be answered. And other studies have shown by using the non-synonymous variant that changes the activity of lambda 4. By using that, they have shown that interferon lambda 3 but not lambda 4 is responsible for inflammation and fibrosis in liver and lungs. But clearly, as you would realize now that comparative studies to understand functions of lambda 3, lambda 4 are lacking, and that was the goal with which we started about 3 years ago in the current project. So we asked the question: can interferon lambda 3, 4 immunomodulate? So what is immunomodulation? Immunomodulation is basically any effect of the protein that we're talking about on immune cells. The presence of this cytokine, does it change the function or the phenotype of the immune cells that it is exposed to? That's the simple definition of immunomodulation. Now there is some data that lambda 3 does change the phenotypes and functions of some of the immune cells, including innate immune cells as well as adaptive immune cells, but absolutely no data was available for lambda 4. Now, why this immunomodulation is important? To give an example, for example, in pancreatic ductal adenocarcinoma, you see that progression-free interval and overall survivability is influenced by the lambda 3 variant RS813. And similarly, in liver fibrosis, the infiltration of T cells is influenced by the variant that changes the expression of interferon lambda 3, but lambda 4 variants do not have any effect on this infiltration. Therefore, this study concluded that lambda 3 is the functional mechanism behind what we see in the genetic association studies. And therefore, clearly, we need to understand in a functional viewpoint how lambda 3 and lambda 4 are different. So we did a simple study where we looked at the effect of lambda 3 and lambda 4 on monocyte-derived macrophages and dendritic cells. The study design is shown here. And we used a commercially available preparation of recombinant lambda 3, which is a mammalian cell-derived one, so it has very good activity. The lambda 4 was a E. coli derived one because that is the only one available commercially, which has a lower specific activity because it goes into inclusion bodies and then it has to be purified and refolded. So the results that I described in the next few slides are already published in JLB last year. So as I said, we started with interferon lambda 4 because some data was already there. Some published literature already said that lambda 3 could immunomodulate. So can lambda 4 also do similar functions? So we basically started with the THP1 derived cell line, and if you treat it with PMA, it becomes macrophage-like cells. So here I'm showing some just housekeeping experiments just to make sure of the preparation is not contaminated, etc. Interestingly, what we saw was the PMA treatment increased the expression of the receptor for interferon lambda, IL-28RA. And therefore, the differentiated cells, but not the raw THP1 cells, respond to interferon lambda 4 by phosphorylating the STAT1 transcription factor as shown here. And of course, if you do a qPCR, a lot of ISGs, interferon stimulated Gs, 5 of them shown here, get significantly expressed in macrophage-like cells. So because the area was also not very which immune cells respond, which do not. So this is an evolving area. So these kind of results were, although not extensive, but they are important to understand which immune cells do respond and which immune cells do not respond. So we continued the experiments. First, we had to determine which point of time do we add these interferon lambdas so that you can see an influence on the immune cells. So firstly, we tried out adding the interferon lambda 4 after differentiation of the THP1 cells into macrophage-like cells, and we added the lambda 4 during activation with LPS. And we saw some inconsistent results that were not really significant most of the times. But once we started adding lambda-4, even during the differentiation, that is when you add PMA to differentiate the THP1 cells, we started to see consistent results. The results are summarized here. Basically, the THP1 cells that were differentiated in presence of lambda-4 showed more pro-inflammatory cytokine expression in terms of TNF-alpha, IL-6, IL-12, not so much with the anti-inflammatory IL-10. But we had to use a high concentration, as you see here, 2 and 6 micrograms per ml. There's a lot of protein added into the media. As I said, it has issues with folding, and I come back to that in a while. But as you see in a different experiment, so 2 micrograms is not really saturating activity of lambda-4. 6 micrograms has a significant increase. So lambda-4 is basically having low specific activity, the one that's commercially available. Just to make sure our experiment was going well, we tested with interferon gamma, which is a strong pro-inflammatory cytokine inducer. So in presence of interferon gamma and LPS stimulation, the effect of lambda-4 kind of fizzles out. So of course, interferon gamma is strongly pro-inflammatory, but interferon lambda-4 is subtly pro-inflammatory. So in presence of interferon gamma, you do not really see the effect of lambda-4. And we did some further characterization, including flow cytometry. So here I see that I show that CD80, which is a pro-inflammatory marker, is increased when lambda-4 is added to the media, but not HLA-DR. So if it's pro-inflammatory, it should be increasing HLA-DR as well, but that we did not see. Agreeing, the qPCR results agreed. TIM3, which is actually an M2 marker. So there is M1 and M2. M1 is the more pro-inflammatory type of macrophages. M2 is the more anti-inflammatory. TIM3, which is an anti-inflammatory marker, seemed to be more expressed when cells were differentiated in presence of lambda-4. And therefore, we said that it's not really pushing towards pro-inflammatory or anti-inflammatory, so we call it the mixed phenotype. And similarly, when we did the M2 macrophage-like cells, not much difference was seen between the cells treated or not treated with lambda-4, but we did see some effects on some genes, important genes like IL-10 and some adhesion molecules. But the important cells are the primary cells. So we did the same experiments, PBMCs. So CD14 cells derived from PBMs can be differentiated into macrophages by treating with GM-CSF, M1, basically M1 phenotype. And then after 6 days, we activate with LPS and then collect the media or isolate the RNA for qPCR. Very clearly, we saw that it is having pro-inflammatory marker expression increase significantly. For example, AT, CD86, HLA-DR, TIM3 is low, so that is it agrees to the bracket of pro-inflammatory or M1 marker, enhanced M1 marker phenotype. But and also CXCL13, but you see that IL-10, which is, as I said, is an M2 marker, is also increased. So the mixed phenotype kind of stays here as well. But certainly, in primary cells also, after the primary macrophages also respond to interferon lambda-4. This is also something that a very new result in the field, because the field was not sure whether monocytes respond or macrophages respond, etc. So again, the DCs also, we confirmed that lambda-4 is working on DCs after differentiation in presence of lambda-4. Several markers were altered as shown here. And again, coming back to PBMC-derived DCs, we see that several ISGs are significantly increased when we add lambda-4 to macrophages. Now, as I said, the field was not clear. We confirmed and clarified this issue. So we very clearly showed that monocytes do not respond to interferon lambdas, but if you differentiate them to macrophages, they do. For example, if you take PBMCs, isolate CD14 monocytes and treat with lambda-3 here, you do not see P-STAT1 signaling. But the same person's CD14 cells are differentiated for 6 to 7 days into macrophages, then they start responding. So the receptor expression is going along with the macrophage differentiation. So, but coming back to the main point that we were interested. So, what is the difference between lambda-3 and lambda-4? So, these protein preparations coming from different sources was a handicap for us, because lambda-3 was having very high activity, lambda-4 comparatively was much less. So, to do a comparative experiment, we had to normalize, these concentrations. So, we did a lot of experiments in number of cells, cell types. And finally, we concluded that about 50 nanogram per ml of lambda-3 and 6 microgram per ml of lambda-4 would give us comparable ISGs, if not equal amounts. So with this, we begin our first experiment on DC. So what is the effect of lambda 4 in differentiated DCs? So we use what is called as a mixed lymphocyte reaction to read out the effect. So we take CD14 cells from 4 individuals and then differentiate them into dendritic cells and then activate them and then co-culture them with CD4 cells derived from allogenic donors. So we use 20 individuals as shown here, and the readout will be the cytokines secreted by the CD4 cells. So we either included lambda 3 or 4 during differentiation of DCs, and then the MLR, and then the results are summarized here. This is a heat map showing the fold change for different cytokines, important Th1, Th2, and Th17 cytokines. Interestingly, lambda 3, although it caused fold changes, but the fold changes were not different between any of these cytokines to say that either it was favoring a Th1 or a Th2 or a Th17 phenotype. But as you see, lambda 4 was causing significant differences between several of the cytokine groups here. But importantly, when you compare between lambda 3 and lambda 4, you see that lambda 4 treated DCs stimulated the CD4 cells to secrete more IL-4, IL-13, which are classical Th2 cytokines. So this was the first functional evidence to show that lambda 4 is indeed proviral. So I told you a lot of genetic studies established that even though it's a very potent antiviral cytokine, in vivo it shows a proviral phenotype, and this is probably the reason, because as you clearly see, IL-4 and IL-13 is increased for secretion, but in asthma, because asthma, one of the endotypes in asthma is an allergic asthma. We had previously reported that older women with at least one allele of interferon lambda 4 generating variant protected these older women from severe asthma. So in that case, if it is promoting Th2 cytokines, it does not explain those results. So that's an open question. But the results shown here probably in this slide probably has an answer for that, where we see that M1 MDMs differentiated in presence of interferon lambda 4 had an anti-inflammatory effect. The IL-1 beta is decreased, but IL-10, an anti-inflammatory cytokine, is increased. But importantly, when you compare the effect of lambda 3 and lambda 4 in the M1 or M2 MDMs that were differentiated in presence of L3 or L4, you see that the lambda 3 specific activity, higher activity, for example, here in TNF alpha and M2 MDMs compared to a lower activity of lambda 4, and similarly in IL-1 beta here. But the most striking result is this one here. So IL-10, which is an important anti-inflammatory cytokine, is increased from M1 MDMs that were differentiated with lambda 4, but decreased IL-10 secretion was seen with the same macrophages derived from the same persons, but when they were differentiated with lambda 3. So this is hinting towards saying that they may be indeed having opposite effects. We could roughly say that lambda 4 could be anti-inflammatory, but not so with lambda 3. So further, we wanted to address the molecular phenotypes of these cells. So the design was basically same. The CD14 cells were differentiated into M1 or M2 types in presence of lambda 3 or lambda 4, and then stimulated with LPS, and then RNA-seq was performed to see the differentially expressed genes. So as this figure shows, about more than 1,000 genes were differentially expressed when the cells were treated with lambda 3, but only about 385 were seen for lambda 4, clearly showing the lower specific activity of lambda 4. When you go to empty MDMs, more than 2,000 genes got induced by lambda 3, but interestingly, very less number of genes, only 39 genes were differentially expressed when the same cells were treated with lambda 4. So here we clearly see that lambda 3 seems to be more active in M2 MDMs, which are called as the resolving MDMs. I mean, in any inflammation, the M2 MDMs come later on, where they take care of resolving the inflammation. So the lambda 3 seems to be active more in these cells compared to lambda 1, which seems to be active more in the M1. That is the more pro-inflammatory. The first type of macrophages that you get in any inflammation is the M1 type. Lambda 4 seems to be more active there, even though interestingly both interferons use the same receptor for signaling. So this scatter plot is more clearly shows that so lambda 3 is more active in M2 MDMs and lambda 4 is more active in M1 MDMs. For example, more than twofold increase in genes in M2 MDMs when they are treated with L3 compared to actually less than actually about tenfold decrease number of genes that are seen in M2 MDMs than M1 MDMs if they are differentiated with L4. Certainly, there is some preference for lambda 3 and lambda 4 for M1 versus M2 MDMs. They do not. At least these results show that they are not the same. They are having different functions or different preferences for these two cell types. And this PCA plot again clearly shows that our differentiation has worked well. This is the M2 group and this is the M1 group. The most differentiated cluster we see the lambda 4 in M1 followed by lambda 3 in M1 again, and the next best cluster is found by lambda 3 treated cells in M2 MDMs, but the lambda 4 treated, they are just looking like the control cells here. NT is no treatment. So next, we wanted to see if the genes are unique or shared between the lambda 3 and lambda 4. And majority of genes, especially if in M1 MDMs, were unique. So that is also surprising because field was not again clear. Some studies had shown that there's not much of a difference in the ISGs that are induced by the two interferons, but there may be differences in kinetics. But another study has shown that there could be real differences if you take the kinetics into account. So, but this study clearly shows that majority of genes are actually unique to the two interferons. So, in vivo, basically, you see both lambda 3 and lambda 4 acting, at least in the heterozygous people. So, we wanted to compare what is the differential gene expression between lambda 3 and lambda 4. So, the results are shown here and here. So, basically, we see about 68 genes more differentially expressed when you do the comparison between lambda 3 versus lambda 4 in comparison to lambda 3 versus no treatment, and about 370 genes become less in M2 MDMs when you do the same exercise. That is at the outset. But if you delve deeper through this three-way Venn diagram analysis, you see some very interesting details. For example, we see that there are about 760 genes shown in green here that become significant only when the comparison is made between lambda 3 and lambda 4, because 99% of the genes are in opposite directions. But they don't show up either in L3 or L4 when they are compared to no treatment. But since they go in opposite directions of smaller effects, they become significant when you do the comparison. It's not surprising that lambda 4 is having a smaller effect because the specific activity is low, but it's indeed surprising that lambda 3 also acts on the same genes, but it has a high specific activity. That activity is reflected in only some genes, but not all genes. And basically, it's a complicated analysis here, but we conclude that interferon lambda 4 can have effects ranging from weak, moderate to high on several of these genes, except for these 534 genes. All of these genes, we can show that lambda 4 is somehow affected. Similarly, except for 103, all of these genes are perturbed by lambda 3. And interestingly, again, you see that this ratio is about 534 and 103 between lambda 4 unperturbed and lambda 3 unperturbed, about 5. It becomes about 60 in M2 MDMs. Compared to M1, it is 5 here, about 60. That is the preference thing, again, is something that we have noticed. We need to work more on this to fully establish this. So where does differences come from? So because lambda 4 is a very difficult protein to work with, no crystal structure is available because E. coli derived protein gets into inclusion bodies, and the folding effects are seen. Folding problems are seen in both mammalian express cells as well as insect express cells. Therefore, no crystal structure is available as yet. But we had an excellent collaboration with researchers from University of Chicago, Juan Mendoza and his grad student Will. They did this modeling for us. Basically, they built a homology model for lambda 4 and compared its interaction with the lambda 3, lambda 4 interferon lambda receptor, which is a heterodimer, basically, interferon lambda R1 and IL-10 R beta. This is a common receptor used by the IL-10 superfamily members also. So the interaction was compared with lambda 3 and lambda 4, and very clearly we see that not much difference in IL-10 R beta, but the unique receptor subunit, the interferon lambda R1, shows very significant differences between lambda 3 and lambda 4. A lot of interactions are missing that we see in lambda 3. So this may be the source of the differences that we actually see in our studies. And if you look at the genes that are affected, very strong effects were seen. I've shown the fold changes here by both lambda 3 and lambda 4 in some of the genes. And interestingly, all of the studies, most of the studies have been associated with some human disease. Mostly, as you see here, the list is full of cancer-related diseases. More systematically using the KEGG or the reactome pathway, we see several infectious diseases and several pathways being significantly affected by lambda 3 or lambda 4. But when you do the comparison of lambda 4, lambda 3, some new pathways come up. For example, the ovals that I've shown the right here, if they are yellow, they show that these are new pathways that were missing from either lambda 3 or lambda 4. If they are pink, they show the pathways that were affected by both lambda 4 and lambda 3. So in the yellow ones, which is the new pathways that you see when the comparison is between lambda 4 and lambda 3, you see human cytomegalovirus infection. There's a lot of data to show that genetic association is in fact there. Indeed there with the lambda variants and CMV infection. And we were surprised to see that Leishmania was also there. And now we have data in the lab to show that type 3 interferons both induce interferon lambdas and they respond to interferon lambdas as well. In the reactome pathway, a lot of TLR cascades are affected and collagen degradation complement cascades. This was in the M1 MDMs, but if you do the same thing to the M2 MDMs, you see a lot of infectious diseases, especially for lambda 3. And when you do the comparison between lambda 4, lambda 3, tuberculosis is the most affected pathway. And leishmaniasis, interestingly, also shows up in the M2 cell type. And if you look at the reactome pathways, extracellular matrix organization is significantly affected because this is the one that is important to resolving inflammation. The M2 phenotype is the one that's required to resolve any inflammation, and we see that significant effect by lambda 4 versus lambda 3 in this important pathway. And also we see cholesterol biosynthesis and some inflammasomes. So with that, I'd like to conclude, our findings. So we have shown that lambda 3 and lambda 4 can both modulate the phenotypes of innate immune cells. For the first time, we show lambda 4 is both proviral at the same time antiviral, anti-inflammatory. And we show that lambda 3 and lambda 4 could have preferences for M1 versus M2 MDMs, but we have to work more on this to firmly establish this. And interestingly, the way we did this experiment has opened up another area, which is macrophage differentiation pathway. There is a crosstalk with the ISG pathways that needs further research. And several molecular pathways, including many infectious diseases, are affected by L3 and L4 in both M1 and M2 MDMs. And the differences in receptor subunit abundance and receptor subunit ligand interactions in the two cell types could be responsible for the effects that we see. We need more work with the more active interferon lambda 4 to understand the full effect. With this, I'd like to acknowledge some individuals. Anand, who was a PhD student, who is now a postdoc, along with a postdoc Manjarika Day, did most of the experimental part of the work, and we are grateful to Juan and Will for the nice modeling work they did, and welcome Trust TBT India Alliance for generous funding, and all these people who have assisted at various parts of the work. And importantly, I would like to thank volunteers who donated the blood for this work. Thank you, and I'll be happy to take any questions.
Thank you, Sridhar, for your excellent presentation. The talk is open for discussion. Are there questions in the chat box, Mohua?
Not yet.
So I will start.
Yes, Professor.
So, Siddharth, I was just wondering, you just touched upon the lambda 3, lambda 4 and COVID. So do you think that lambda 3, lambda 4 response to the variants are likely to be different?
The effect seems to be small. That is the problem. So if the effect was really high, we would have seen much higher impact publications come up already. But I also heard that they are in the works. So we have to wait a little more time to see if really lambda interferons, the genetic variants, have a bigger effect on either the susceptibility or the pathogenesis on the severity of the disease.
Exactly. Symptomatic cases and the severity can be connected or not.
Yeah, I suspect it may be playing a role in the severity rather than the susceptibility per se.
Right. Severity might be a
yeah.
Good.
We have a question from Dr. Arvind.
Uh-huh.
So he's asking what role IFMs could have in resolving phenotypes of macrophages in general, particularly in TAMs.
Particularly in?
TAMs.
TAMs.
TAMs, such as tumor-associated macrophages. It's a very interesting question. As you see, a lot of cancer pathways have come up in lambda 4. If I can show you again, it's still there, I guess. Can you see this? Can you see the slides again?
Yeah, it's still there.
Okay. If you see in the M2, even though we see only 39 genes were affected, can you see this slide, the pathways?
Yes.
Yeah, so lambda 4, even though only 39 genes were affected significantly, you see a lot of cancer pathways coming up significantly here. Even though the significance is less as well as number of genes are less, but you see glioma, melanoma, another pathway on cancer, and prostate cancer. So his question relating to TAMs is interesting because, as I said, M2 macrophages are also the lambda 3 seems to have a preference for M2s, but lambda 1 does not have. We don't know how this would play out. You have to do either a genetic study or you have to characterize the phenotypes and then correlate with the genetic variants after isolating these TAMs from tumor tissues. It's very interesting. There is a lot of, I would say, there will be a lot of information, but how much of it and how to get that is also an issue because you need a larger study to have sufficient number of people to do the get a power for your study. So that's interesting because the data shows it's a lot of cancer pathways are associated, and therefore both in M1 and M2. So M2, as I said, this may all be affecting the phenotypes of the TAMs. It is known that lambda 4 is kind of, if you have lambda 4, it is risk for cancer, prostate cancer and ovarian cancer. GWAS has shown for ovarian cancer. So this is consistent with the antiviral effect. For antiviral effect, you need Th1 response, and also for anti-cancer effect, you need a Th1 response. But the lambda 4 is more of showing of Th2 kind of phenotype, but definitely it has a lesser inflammation. So in other words, you are at risk for some virus infections, but you are for virus infections and cancer, but you may have a protection from some inflammatory diseases. So that really is not clear, but it appears to be like that.
Yes. Anupam?
Yeah. Sridhar, it's a nice talk. I just one question now in the context of the TAM and your tumor and the M1, M2. Now, most of the papers coming out right now in the cancer biology, we cannot basically differentiate M1 and M2 state basically in the cancer's condition also, as you agree for that. My question is that in normal infectious condition, can you differentiate either genetical signature or transcriptome signature M1, M2 state? That's my question.
Yeah, thank you for your comment. But see, if you look at how the M1 and M2 are made, firstly, so M1 comes first. So when the inflammation starts, it's obviously the macrophages will become M1 first. You won't see M2. M2, the same macrophages are converted to M2. So your question of can you identify M1 or M2 in the same point of time is difficult because M2 comes from M1. After the inflammation is high, to resolve the inflammation, the M1s gets to differentiate into M2, getting a lot of signals from neutrophils and other cells.
Means polarized, this is a complete polarized state or mixed?
Yeah, so it polarizes M1 and then M1 completely disappears, and at the end of the wound, for example, tumor they say is a unresolved wound, right? Unhealing wound. So therefore it's only you see M2s there because it's like a wound. The immune system thinks it's a wound, so it's trying to heal by having more M2 there. M1 won't there. M1 is there. It's good. You'll eliminate the tumor probably at the initial stages, but when the tumor is fully formed, M2 is the one that dominates because the immune system thinks that it is a wound. It's basically a wound, and it's trying to heal the wound rather than be aggressively trying to eliminate the wound. So it's difficult to find both M1 and M2 at the same time.
Yes. It's mixed basically.
Right.
Yes, Professor Parthuda, please.
Sridhar, I just wanted to make a comment on your observation. Correctly so, I mean, like you mentioned that M2 gets, M1s get converted to M2, but you also mentioned that at the end, you will only find M2s and no M1s. In many cancers, that does not seem to happen. Where there has been time series sampling at the single cell level, it turns out that you find both M1 and M2s even after a long period of time. And that's probably because there are other proteins from the tumor that's kicking in or from the immune microenvironment that's kicking in and preventing M1s being converted to M2s. Reasons are unknown, but both M1 and M2s can coexist for a very long period of time.
Yes, I do agree, but there may be a difference in the ratio. So I suspect because tumor exists because there is no M1, there's no sufficient M1. M1 will keep coming from the newer, probably the monocytes that come in and start to because this is a continuous process. So there will be stimulus for the monocytes to continuously becoming M1, which becomes M2. But the ratio of M2 probably I suspect will be more. I don't know.
There is a question from Dr. Saroj. He is actually asking, like, following up on the COVID question, if you can comment on the IL-6 pathway.
What is the exact question? My comment on the IL-6 pathway in COVID.
Yes. Yes. So following up on the COVID question, he's saying. So I mentioned about the reactome panel. So in relation to that, he's asking about, like, if you can comment on the IL-6 pathway.
Yeah, I'm aware of his work that he showed that IL-6 is important and netosis is also related to that. Well, we know that IL-6 is a damaging cytokine, and that is that you obviously see that as one of the cytokines in patients in ICU. Yeah. So the question is why you see IL-6, which is a pro-inflammatory cytokine, is supposed to be pro-inflammatory cytokine, which is acting detrimentally to the host. Obviously, people in the ICU are under severe disease, right? So why IL-6, which is supposed to be protective, by eliminating inflammation or infection, is actually detrimental to the patient. This is the result of the cytokine storm issue, which is a much bigger question. So probably the virus is not detected until a lot of replication and infection has happened. So at that time, the body kind of responds in a very complicated way, thinking that things are gone beyond the control. So the cytokine storm, I don't know the trigger for that. So that kicks in, and IL-6 is part of that bigger pathway, which no one of us really understand. The cytokine storm pathway, that really needs understanding in COVID, and septic shock as well. The same thing happens in septic shock.
Okay, if there are no more questions, I hope there are no more questions, Mohua.
No more questions. Yeah, okay. Thank you.
Okay, yeah. Thank you, Mohua, for handling this so well. And thank you, Sridhar, for your excellent presentation.
Thank you.
It's been a long session starting at 10 o'clock till 1. And I thank all the speakers of this session, including Professor Majumdar, for inaugurating the program and making his inaugural address. And we now need to break. We'll come back. And I invite all of you to come back and join and participate in Professor Soumitra Das's talk. And with this, I hand over to Anushwa for her announcement. Thank you.
Thank you, Professor Sengupta. Thanks a lot to our distinguished speakers for these extremely interesting and illuminating talks. And also thanks a lot for sharing the anecdotes of your association with Professor Das. So we'll take a break now, and we will reconvene outdoors at the lawn at 2 p.m. Thank you.
May I also take this opportunity to quickly mention that lunch is arranged in the cafeteria. So everyone please get your lunch box for everyone who is attending. Thank you. And I'll see you at 2 in the lawn.